All right. 2024 Parasitology test one. A word for the day. You can only live when you die. Die to your flesh desires, die to your addictions, die to pride, and allow the Most High to give you a new life worthy suffering for. You you aren't too wasted for Jesus to pick and make a wonder to the world out of your broken pieces. Remember that when the time had fully come, he came and died for you and me. So, question number one, produces rectal prolapse in heavy infection. Such questions. How does a question just start like that? And my voice clear enough? Like properly clear? All right, thank you. So, produces rectal prolapse in heavy infection, especially among children. Imagine a question that starts just like that, produces. So when it comes, every time you hear of rectal prolapse, think trichuris, trichuria. Now let's say just something about trichuris. Anyone that can just mention anything that they know about this parasite? Anyone? All right, thank you. Anything else? As a thread way? Anything else? Thank you so much. Is this the one known as a threadworm or whipworm? Or maybe both names? Thank you. So it's a whipworm. The thread is that ancylostoma. The two, there's ancylostoma and the strongyloids. One of those should be a threadworm. Okay, thank you so much. So trichuris is usually associated with, Doc, what did you say again? All right, thank you. Usually associated, yes, with Ascaris lumbricoids. And this guy is known as a whipworm because of the appearance. You know, most of the worms, if not all, when you look at their morphology, the females are usually larger than the males. And when we know that when you're talking about the classification of worms, maybe we can just talk about that now so that the other questions are going to be okay. Let me not move ahead. When you're talking about classification of worms, you know you can classify worms into two. You have got the flatty worms, which we also call platy helminths. So these, remember, we call them helminths. And we are saying for the helminths, you can classify them into first nematodes, which are the round worms, and then you have got the platyhelminths, which are the flatworms. Now, nematodes, you can classify them depending on the location. You have got the intestinal guys, so you can say you have got intestinal helminths, and then you have got also those which are in blood. And then for the platyhelminths, the flatworms, you can classify them into cystodes or trematodes flukes, right? So that's a basic classification. And when you're talking about trichuria, it's a nematode there. What more should we know? Just maybe know that it has got a thick posterior end than the anterior. And when you look at how where it resides, the adult worm, you are going to find the adult attached to the cecum or the colon or the appendix. You can see that it's a lot, right? Cecum, colon, appendix, and also the anal canal. Basically, the ends, the large intestine is the anal canal there. And it has got only one host, and that is man. I think that is of importance. Another important thing to know is what is the infective form. The infective form is L3. So when you ingest eggs which contain L3, that will be what do we call L3? That is the because L1 is a labyrinth labyrinth form, and then you have got L2 because when L1, this is the larva, is going to molt number one, and then it is going to molt again, changing, and then it's going to go into the filariform larva. So that's the infective form. Okay, basically that I think I've mentioned enough about this guy. Which of the following statements is the correct life cycle of Trichomonas vaginalis? Trichomonas. Okay, let's mention again something about Trichomonas. The first thing first that we have to know about this guy. Let's hear from anyone. Anyone that wants to just anything that they know about Trichomonas. Thank you so much. That's a basic thing that we should all know. It is an STI, sexually transmitted infection. And when you look at the location is going to, because it's an STI, you are going to find it for females is going to be, it will reside in the vagina, for males in the prostate gland. For both, you are going to find it in the urethra. So you can now think of the diagnosis. If it is in males, if it is in female, so you can use urine, right? In both. In females, you can as well, because it's an STI, you can as well look at a swab. So besides that, one thing we need to know is that it's an obligate parasite. It doesn't just stay in, and importantly, it infects only squamous cells, not columnar cells. That's important thing to know about Trichuris trichuria. It is going to infect squamous cells and not columnar cell. Let's say this is the guy like that, and say this is the anterior aspect, and then let's say down, this is the posterior aspect. So this guy, another thing that you are going to be able to see is that it has got flagella, and it has got four anterior flagella like that, and then it is going to have one posterior flagella, but it doesn't, the posterior one doesn't reach to the end, like it's short, just like that. And then you are going to have one anidulating membrane. It goes like this. You have got, it looks like a flagella, this membrane which goes like this. It follows the plasma membrane like that. So these are the four things: four anterior flagella, one posterior, and one undulating membrane. And what else should we know about this guy? It has got a trophozoite. I think that's the infective form, the trophozoite. About other forms, I don't know about any other form, but the trophozoite, which is going to also act as the infective form. Another important thing we need to know is that it is going to divide by longitudinal binary fission. And of course, infection can be transmitted from the parent to the mother. And such infection, where it comes from the parent to the mother via the placenta, that's known as vertical transmission. Please take note that this guy divides by longitudinal binary fission, but infection can be transmitted from mother to child via vertical transmission. Okay, and then it has got some virulence factors. What are the virulence factors? It's going to have the protein ligand or protease. And the aim of this guy, protease, you can think of a protein, right? From the word protease. But not normally as you know it, protease breaking down proteins. The protease here is going to be used for adherence to surfaces. Besides that, another virulence factor that this guy has is lactic acid or acetic acid, and those are there to be able to lower vaginal pH. And then another thing is that it has got what is known as cysteine protein, and the cysteine protein is going to be is for hemolysis, breaking down red blood cells. But the thing is that this guy loves an alkaline pH. So when it goes to the vagina, it's going to stay there until the environment becomes nice enough, and then it will be able to raise the pH to a point where it can stay for it to mature and multiply. But remember, the lactic acid that it has is to lower the pH. Lowering means you are making it more acidic. Okay, hope that's okay. So back to our, yes, please. Was that a hand? Okay. You can see our answer here is trophozoite. So which of the following statement is correct about Trichomonas vaginalis? Trophozoite transmitted sexually, reproduced by longitudinal binary fission. Trophozoite develop and then transmitted sexually. Look at a cyst and trophozoite transmitted sexually reproduced. So about cyst, no, it's just a trophozoite, which is the infective form. That is the one that is going to be transmitted and then it's going to undergo division. And then B cyst, again there is a cyst there. D trophozoite transmitted sexually. It's not transverse binary fission but longitudinal. So that is that. And then number 3, Trichomonas vaginalis reproduced by transverse binary fission. Of us, pardon, there's a question. Yeah. Okay. Yes. So you know, the pH for the vagina where this guy is going to go is a bit more acidic, but it needs a slightly higher pH, a slightly alkaline pH. Now, this virulence factor that it has, the lactic acid or acetic acid, it lowers pH. So I was also trying to think about the science there. Maybe I didn't take much time to discover the actual science, but is the best environment for it is alkaline. Actually, it's going to raise a bit the alkaline pH when it enters. But this virulence factor, specifically the lactic acid or acetate, that is a function. It lowers the vaginal pH. It lowers pH. So I was also trying to look for that reason because they don't seem to add up. Yes. Hope that's okay. All right, thank you. So Trichomonas vaginalis, we've said that A reproduced by transverse binary fission. Is this true or false? It's false, right? We said longitudinal. And then B, thank you. B, it is an invasive parasite that adheres to the squamous epithelium. Okay, about squamous, that is true, but about invasiveness, that's a lie, right? That's a lie. It is not invasive. And then C, its virulence is due to protease which help in adherence, lactic acid and acetic acid which lowers the vaginal pH, and we've just said this is true, right? And then D is associated with vulvovaginitis that is characterized by non-severe inflammatory response. This inflammatory response is severe, not non-severe, so it's gonna be associated with vulvovaginitis. Actually, when there is infection by Trichomonas vaginalis, it will start going a bit on the surrounding tissue. And you know the surrounding tissue, you have got the vagina. There's going to be inflammation of the vagina and some other tissues nearby. Number 4, which of the following statement is correct? Is the correct life cycle of Taenia solium? Okay. I want to ask this question about Taenia. Anyone that can just say anything? Anything that they know? I've seen a hand. Anyone? Just anything. Basically anything. Okay, there's a question. We can start with a question before we can go on. So question 3, the answer is C. Okay, so back to question 4. Anyone that can say anything that they know about Tinia sodium. Just anything. Small intestines. Thank you so much. That's one thing we shouldn't forget. Small intestine. Besides small intestines, between a pig and a cow, which one do you think about when you hear sodium? The pig, right? Thank you so much. So Tinea sodium is also known as pork tapeworm. Remember this guy? It's a cystod. Let me now mention this. I think this is the right time for me to mention these guys so that the rest of the questions are going to be interesting. How do we classify helminths? So when you're talking about the classification of helminths, you can number one classify them looking at the mode of infection. Mode of infection. When you are saying mode is how are we going to get them? And on the mode of infection, there are a lot. Number one, we can get them by chewing them, by getting them in food. That is ingestion, not injection. Ingestion. We know injection is via blood, right? This is ingestion, oral fecal. And for these guys that we can get by ingestion, we can either get the eggs or we can get the larva. I've left out ara there. Or we can get, sometimes the larva can be in a cyst. So we say encysted larva. So you have got this, let's say, is your cyst, and then inside you have got a larva. It's a small worm. So we can also have the encysted larva, like that. Please take note, those helminths that you can get by ingestion through the mouth, but it is through their eggs. You have got Ascaris, Enterobius, and Trichuris. So we can abbreviate them. You know the word eat, right? Since we are getting these by these eggs. So we can abbreviate the three helminths by a simple thing, eat. We are eating the eggs, that is ingestion. So Enterobius vermicularis, Trichuris, Trichuria, and then Ascaris lumbricoids. And then for the larva, this is the larva with intermediate host. So it has got an intermediate host. Not every worm has got an intermediate host. Some of them, they just come in our bodies direct. And this is one which is known as Drancuculus medinensis. When I was reading about this guy, the Drancuculus, that should have been, which one is which? Mednesis was a place and then Drancuculus was a person. Yeah, should be. There's that combination. So think of this is Drancuculus, which we get the larva which has got an intermediate host. And then for the encysted larva, we have got what we call Trichinella. Please, this T is for Trichuris. And then for the encysted larva, it is it Trichinella. Besides us ingesting these guys, we also have those that can penetrate the skin. We don't get them through food, but they enter the skin. And here we have got two important guys, the hookworms and the strongyloid how do you call it? Stacularis, right? The strongyloid stacularis. You know the hookworms, we have got Enterobius, and we also have which one? Not Enterobius, but Neceta americanus and Ancylostoma duodenale. Those are the two hookworms. So these guys will basically come and enter the skin, and then they will infect us. So besides those which enter the skin and get ingested, we also have those which are going to come via blood-sucking insects. And blood-sucking insects, what do we call them? Basically arthropods, right? Like mosquitoes. So mosquitoes can be able to transmit these worms, and we call those as filarial worms. Filarial worms are basically helminthic infections that are transmitted by arthropods like mosquitoes. And these guys, when you see the chima it can, some of them enter the lymphatic system. Others, we are going to be able to see them on the skin. Literally, they are clawing under the skin. You can see how they are moving. And then another mode of infection is inhalation. Like, you are going to inhale them, you are going to inhale the eggs. And for those that you inhale, you can see the same guys that you ingest, they eat. So you have got the Ascaris and the Enterobius. Except Trichuris, you'll not be able to inhale it. So this is on the mode of infection. Besides mode of infection, you can classify these guys also looking at their ability to lay eggs. So some of them, they lay eggs like chicken, right? And then they'll be able to infect. Now, of another important thing that I want to mention here is something easy that we have already mentioned a bit. And then, so here, helmets, as we already said, that we can divide these helmets into two. We can divide them into the flatworms, which we call platyhelminths, and then we can divide them also into the nematodes, which are the roundworms, nematodes. When it comes to the platyhelminths, we said we can divide them into cystodes. We know cystodes are also known as tapeworms. That's where we have got the tinea. And then we have got trematodes, which are also known as flukes. So when you look at the cystodes, we have got things like tinea, Echinococcus granulosus, Diphlobothium latum, those interesting guys. And one important thing that I want us to remember about these guys is that, let's start with nematodes. Nematodes are very good guys in that they are unsegmented. You not see them like this. They are not like this. Remember, these are roundworms. They are unsegmented. Besides that, they have got an inner body cavity, like the entire GIT, they have got it. Besides that, they have got separate sexes, meaning that a male is a male, a female is a female. You are going to understand what I mean by that. But basically, female is female, male is male. It's not the same worm with the two different sexes. Besides that, another thing about nematodes is that they don't multiply in the host. Please take note. They don't multiply in the host. So any nematode that you think about, they don't multiply in the host. What other thing? Another thing we need to know is that both the adult and the larva can produce injury for the nematodes. And those guys that produce the intestinal guys, they can either be intestinal or blood. So you have got those that can be found in the intestines and those that can be found in blood or tissue. Obviously, those which are found in blood are going to be more systemic, right? So that's another thing we should think of. And when you talk about nematodes, the effect of the parasite depends on the type of species. Some species have got a bigger effect than others. The intensity of the infection. If you have got more worms, the more the problem. Another thing is the location. So some locations, the infection might not be that bad, and some locations it will be very bad. Again, in nematodes, we have got the intestinal guys, as we said. Let's now divide them into like that. So intestinal and those which are non-intestinal, so those which you find in blood or tissue. Now, for those which we find in the intestines, they are actually further divided into soil transmitted helminths, meaning that they come from the soil and then they'll be able to enter the GIT. And then you have got the non-soil transmitted helminths. For those that are transmitted in the soil, we have got ascaris. Please, this is important. I don't know how you get a lot of questions. Even just this paper has got a lot of questions. Just this classification. So those that are gotten from the soil, we have got ascaris lumbricoids. We have got Trichodis trichuria. We have got a hookworm, which are the Ancylostoma and Necata. And then we also have the strongyloids, stacholaris. I want to use a mnemonic. I wanted to make a mnemonic something like tash. You can see like that. Since this is like, it's a bit similar, right? STH. And then here is also STH, just that there's also an A. Ancylostoma, ascaris, and then trichuris, and then you have got the hookworms, and then you have got the strongyloid. For those that are non-soil transmitted, you don't transmit them in the soil. You have got enterobius, vermicularis, and the trichinella. Now, of all these things that we have mentioned here, only two are going to infect the large intestine. Only enterobius vermicularis and the trichuris trichuria. The rest of them will infect the small intestines. Think of the enterobius is the one that moves out like it. At night, it will come out from the anus and then it starts looking. In the morning, it goes back. Trichuris, large intestines. Sorry, enterobius. And also trichuris, also large intestine. The same guy that causes rectal prolapse. Just remember the rectum, right? That's large intestine. Okay, I think that is gonna help. And then when you come to those guys that are spread in blood, you have got filarial worms and drancuculus. The same guy that we said here that it is spread by the not the encystic, but it has got an intermediate host. So drancuculus and the filarial worms. The filarial worms are found either in blood or tissue. And this is where you have got some which are lymphatic filarial worms like Wuncherella, Brogia malay, and those others. And then others are subcutaneous like lower, onchocerca, Mansonella. You are actually going to see them on the skin, just beneath the skin. You can even see them when they are moving. So those are subcut. What should I mention again? Okay, cystodes. Another thing I should mention about cystodes, just cystodes, they have got no GIT, they have got the head, and their head, please take note, the head of cystodes. And another thing that I didn't mention, the nematodes don't have these things known as suckers, hookers, and those things. They don't have. Cystodes have got a head which has got suckers and hookers. And then they are going to be divided. You are going to have the head, which we call the scolex, the neck, and then you are going to have the strobilus. The strobilus is like the entire tail. So you have got the head, which is the scolex, and then you have got the neck, and then you have got the entire tail, the strobilus. And this strobilus is going to have proglottids. Those proglottids, you have got like 1,000 of them. Those are going to contain eggs. For example, Taenia solium, they contain 40,000 eggs. Each proglottid contains 40,000 eggs. So that is important to know. Another thing that we need to know is that these guys are monoecious. The meaning of that is that they have got the same worm is both female and male. It has got the reproductive organs for male and female. Another thing you need to know about both cystodes and trematodes is that they lay eggs. The word for laying eggs is oviparous. Both cystodes and trematodes are oviparous. Now, the difference between cystodes and trematodes is that for the trematodes, they have got an incomplete GIT. The cystodes are going to have, remember we said that nematodes, which are the roundworms, have got a complete GIT. And then for the cystodes, they have got no GIT. The trematodes, they have got a GIT but it's incomplete. They have got a mouth but they don't have an anus. So it's incomplete. And another thing is that the trematodes have got suckers on the head but they have got no hookers. Different from the cystodes which have got both suckers and hookers. So that is a very nice thing we need to take note. And then the last thing, I think I would have talked about everything that I wanted to be talking about on each question. So we'll just be answering the questions. When you talk about the ability of these nematodes to how they will infect, some of the nematodes are oviparous. The meaning of that is that they are going to be able to lay eggs, and those which are oviparous, you have got some which can lay segmented eggs, others the eggs are unsegmented. So for the unsegmented eggs, you have got, I've just used the simple mnemonic so that you know all the oviparous. Tana. So you have got the T there being the Trichuris trichuria. The A is the Ancylostoma duodenale, and then the N is the Neceta americanis, and then the A is the Ascaris. So all these guys here, they are oviparous, they lay eggs. But all these Ascaris and Trichuris, these two, Ascaris and Trichuris, they have got unsegmented eggs. And then you have got some which are viviparous. For viviparous guys, they produce larvae, not eggs. It's the larva. And here you have got basically think of any other worm other than this tana. But before then you have got a combination of oviparous and viviparous, and that is known as the ovoviviparous. And you only have one that you need to know here is the strongyloid. So if you know that strongyloid is ovoviviparous and then tana is oviparous, the rest of the worms are going to be viviparous. They lay uh, they are going to lay not worms, the larva. Okay, basically that. So back to our question number 4. Sorry for speaking too much, but that is going to answer a lot of questions ahead. So the way Taenia solium infects human is that, specifically this is Taenia solium. We are getting it from pork. So what happens is that, yes, you have got a man that has got niasis. So man with the infection. And this man with the infection, what is going to happen? You know, that infection, the tinea they have inside has got proglottids. And those proglottids, those are basically segments which contain eggs, like 40,000 eggs inside each one of them. And you have got like 1,000 proglottids. So you can think of how many eggs are there. So what these guys will be doing in the man is that the proglottids are going to start to detach from each other because they are part of the worm. So they'll start separating from the worm. So detachment of the proglottids. And when these proglottids begin to detach, what will happen is that they'll now start to be shed into feces. And when they are shed into feces, what happens is that the eggs are going to be discharged in the feces. Okay, because the proglottids themselves contain the eggs. So the eggs which have been discharged in feces, they have got two different parts. Number one is that a second man can end up getting those. Okay, the second man can end up getting them, and that can be via dirty water or such, right? But not necessarily water. The pig itself is normally the one which is going to come and play with the dirt and then get it, and then it will go into the pig, and then like the second man can eat the pig and then get the infection. But let's say we're talking about the pig, so the second host is going to be able to ingest the eggs. Let's say it's the pig, or let's say for whatever reason it's the man that has ended up getting those eggs. So those eggs are now going to be able to grow, and they'll go basically in the large intestines first. Okay, and when they go there in the large intestines, they are from the large intestines they are going to move and go to the blood, and then from the blood they can go to any part of the body. Now the eggs, before we shed them in the feces, they can actually also undergo what is known as external autoinfection. What they'll do is that they'll go back and infect the same man. They'll penetrate the large intestine and go back into circulation, infecting the man again. That is external infection. But you also have what is known as the internal autoinfection. Internal autoinfection is basically where the eggs, which were present in the large intestines, can now undergo reverse peristalsis. They begin to be pushed back into the stomach of the human being. They cause again infection. So please take note. That's how infection can happen. So we are saying that the life cycle, option A, embryonated eggs are ingested by the human, and then oncosphere is going to hatch in small intestines and then penetrate intestinal wall, circulate to the muscles. When they go to the muscles, they are now going to cause cysticercosis in muscles. They can as well go to the brain to cause cysticercosis there. If you look at the other option B, embryonated eggs ingested by human host and cause fear hatch in large intestines, penetrate penetrate intestinal, circulate. Actually, it's not supposed to be large intestine. So where you see large intestine, that's out. And then this saying embryonated eggs ingested by human host oncosphere hatch in small intestine, penetrate intestine, and they don't penetrate the liver. So, of the explanation I gave, when they reach the large intestines, in the large intestines, they can cause autoinfection, at least from the nose that I was using. But hatching is happening where you are removing. You remember you've taken this egg. We can't see the screen. Are others able to see it? Oh, it should be network that side, doc. Okay, so you can simply disconnect and try to connect back. I think it will show back. So please take note that when these guys reach like the large intestines, they can undergo external autoinfection. But the actual arching of the oncosphere is happening in the small intestine. So A is correct. And the number 5 is 2 is a specimen of choice for the diagnosis of infection caused by capillary penis and then trichuris trichuria, trichinella spiralis. So here you can see the answer there. Anyone that knows how is trichinella spiralis diagnosed? Anyone that knows that? I was saying it should be like muscle. Yeah. So for the stool, you can use this capillary and the remember we said it's part of the intestine. Pardon? All right. Thank you so much. Number 6, in Plasmodium falciparum, infection, seasons rupture to release. Previous question, it's A and C are correct. So option D, Capillaria philippinensis and Trichuris trichuri. So a question on Plasmodium falciparum. Again, on Plasmodium, this is something that I believe we all know. So this is a Plasmodium. We have got these, of course, five different species we can talk about, but four are important. We have got they can cause different infections. like the different forms of the falciparum. You have got the vivax. Let me first mention the infections that they can cause. You have got a benign tertian malaria. You have got the malignant tertian, and then you also have the benign qua- quartan. Now, which species of the Plasmodium cause a benign tertian malaria? Anyone that can help with that? Thank you so much. Just like normally, when you see vivax and ovale, they normally move together. Just like both of them, they can cause relapse because they form the hypnozoids. But for falciparum and malaria, they are involved in recrudescence, meaning that we don't kill them completely. So the infection is going to come back again. These ones cause relapse because of the hypnozoids, which can remain dormant in the liver. And you need to take note that when you have got infection of the liver, you cannot detect that until the merozoites are released into blood. That's when you can detect the infection. So someone can have malaria, but then you are not able to detect it because the infection is still in the liver. And then for the malignant, this is where I've got falciparum, and then the benign quartum. This is the malaria. And one thing we need to know about these guys is that for these three guys, these ones are going to have fever that happens every 3 days. And then for the malignant, for the benign tertian, the fever for this guy happens every after 4 days. Another thing we should know is that for the falciparum, you have got it infects red blood cells of all ages, whether old or small or young. And then the morphology, you have got a trophozoite, which is the ring form. Please take note. A trophozoite is a ring form. And then the seasons seasons can rupture. When the mosquito bites us, it gives us the sporozoids. Those sporozoids go to the liver where they are going to be able to change, going and forming the seasons. Seasons basically have got merozoids. So these guys can rupture and release those merozoids. To be specific, the falciparum can release 32 merozoids in a season. And then you have got the gametocytes. Gametocytes for falciparum are banana shaped. It's also known as a crescent. For the rest, the gametocytes are round. Please take note of that. You have, of course, other forms. Vivax, Vivax, this guy causes infection to the young red blood cells. Just think of it. V and Y are letters which are very far in the alphabet. So this one infects young guys. And what you are going to see is that the season is going to release 8 to 24 merozoids. And then for ovale, it infects also young. Remember we said that mostly where you see Vivax, also ovale, because they have got similar characteristics. That's why they both have got relapse. And so ovale is just that for ovale, remember O comes first in the alphabet than V. So also the merozoids are a bit fewer. It has got only 8 up to 10 merozoids. And what you should know about these guys, ovale and Vivax, they show what is known as the I don't know the pronunciation of the word scarfna scarfna dots, something like that. So these two guys have got these dots that you'll be able to see in their structure. And then for the malaria, it infects odd cells, and it shows like 6 to 12 merozoids. It's actually also a small number, 6 to 12. So we can see that Plasmodium falciparum is 32 merozoites. You know that malaria is very bad. It can cause recrudescence even after 40 years. That's very bad. Okay. The following parasites is or are correctly matched to the vector. Onchocerca valvalis, mangofly, Lora loa, Chiros Chilosops species, Honcharella bancrofti, Colicoides A and B. So as we can see there, lower is going to be transmitted by Chirosops species. When you talk about Honcharella bancrofti, this guy is transmitted by mosquito, specifically of the genus Phonestas. Phonestas. And for the Phonestas is this word. And also there's also the Gambiesi. That's a mosquito, Anopheles mosquito. So those are the genus. And then for Onchocerca is spread by the black fly. Take note of that. So specifically of the genus Imutium. Okay. Number 9. Which of the following has the potential to produce? I've left out. Oh, question 7. The most common cause of visceral larva migraine. Ancylostoma branziriensis, Enterobius vermicularis, Strongyloids stacularis, Toxocara canis. Yeah, for the this is visceral larva migrants. Most of the worms that you are going to see, like the filario worms, they mostly cause cutaneous larva migrants. Most of them, where the worm is moving around the skin. But this T, just think of T tissue. The Toxocara canis is, I think, the only one that I've seen that causes visceral. Because majority of these other guys, Havena lower, these guys are going to be causing cutaneous. And you know the ancylostoma guys, these are basically other worms that are causing other infections. Number 9, which one has the potential to produce autoinfection to man? We should have mentioned this already, or if I didn't. Autoinfection, strongyloids stacularis. This is one thing we should know about them. Every time you think of autoinfection, think of strongyloids. Of course, you have got those others where they are going to be, like for example, is that the vermicularis, which causes retroinfection, meaning the worm can go back. Enterobius vermicularis. Strongyloids. Always think of autoinfection when you think about this guy. Or maybe I can ask, how is the strongyloids infection? How does it happen? Anyone that can give a brief something on that? Okay, I'll mention it a bit in front. Number 10, what parasite or parasites are which parasite or parasite has a blood lung phase? Yes, please. Okay. Yes. Mhm. Thank you so much. That's a good quick summary from the doc there. We said the strong law is ovoviviparous. So once it enters the body, you already have the larva there, and then the larva is going to go on causing infection. Number 10, what parasite or parasites has a blood lung phase in the life cycle? Blood lung phase. And for these guys that travel or move to the blood and then to the lungs, you are going to see them associated with also Loeffler's syndrome. So Ascaris is one of those guys. Strongyloistecoralis. Any other that you know that causes Loeffler syndrome? Anyone? Okay. Besides Ascaris and strongyloids, also the hookworms. You know the Ankylostoma duodenale and the Neceta americanus. Those guys as well are involved in causing Loeffler syndrome because they go to the lungs. Okay. Is there any other contribution I'm hearing? Hands? Yes, please. Thank you so much for that beautiful one. Hope we've gotten that. So this mnemonic nut, they have got a blood lung, except the T. These guys are soil transmitted, except the trichuris. The trichuris is also soil transmitted, but it does not go to the lungs. Okay. Thank you so much for that. Number 11, nematodes pathogenicity is dependent upon the following except. We already mentioned this isn't its species, intensity, location. So except incestation. Incestation is ability to is being transforming to form a cyst. Number 12, regarding Trichomonas vaginalis parasite. Both cyst and trophozoite are the infective phase. We did mention this that the infective phase is the trophozoite. It is an obliteratory parasite that infects the columnar cells, and we said it infects what kind of cells? The squamous cells, thank you. And then see, it lives in the reproductive and urinary tract of human with high infection rate in male. What is wrong about this statement? Yes, the infection rate is high in females, not in males. It is pariform flagellate, possessing four anterior flagella and one undulating membrane. We know this is true, right? Plus, of course, it has got also a posterior flagella, which is short. Okay. 13. This is true about Cryptosporidium parvum. Cryptosporidium parvum has zoonotic potential and infection being acquired by ingesting oocyst with two sporozoites. Inhabits the brush border of mucosal epithelium, stomach, ribs of large intestines, villi of small intestines, rectum, appendix, liver, pulmonary tree. Mature oocysts with four sporozoites are the infective forms. Oocysts are stained by the zinc stain as they are non-acid fast. So I can mention something real quick on here. When you are talking about Cryptosporidium parva, number one we should be able to know is that it is a worm. That should be easy, isn't it? So what is the point of infection? So Cryptosporidium is going to infect the brush border cells, the brush border of the mucosa epithelium. You know, the brush border is, you have got these cells like this. So just there, it will be infecting that. And specifically, it is infecting the brush border of mucosa epithelium of the stomach, the crypts of the large intestines, the villus of the small intestines, the rectum, the appendix, and the pulmonary tree. So you see most of the GIT, including the appendix, like from the stomach to the large intestines, including the appendix, also the rectum and the pulmonary tree, just except it doesn't go to the liver. So that's why we are removing option B, because it's also including the liver. Another thing we need to know about this guy is that it has got an oocyst, and the oocyst of crypto has got four sporozoites. The difference with Isospora berry is that the oocyst has got two sporocysts. That's for Isospora berry. This one, the oocyst, has got four sporozoites. The other one, oocyst, has got two sporocysts. Okay, hope that's okay. Another thing we need to know about this guy is that you can use the Zenith Nicel, the ZEU Nicel stain, which is a ZN stain, but it's acid fast. So D is not correct because of the not acid fast. And then another thing is that in water, these guys can actually, the horses can survive in water even when you put chlorine there. And the way they are spread, the crypto is spread via fecal oral. And autoinfection is possible for this guy. Take note, besides the strongyloids, this guy has got also autoinfection. And just like others, worms which are not nematodes is also monoxenous. It has got, man is usually the suitable host, one host. It has got one host. That's the monoxenous. But it can act as a reservoir. It can be found in some other animals acting as a reservoir. Even man itself can be a reservoir, cats and dogs, even cattle. So about the life cycle, let me mention it because there's something long life cycle that is in front. So what is going to happen is this: for crypto, when we ingest the host is going to travel to the small intestines. And then when it goes there, what is going to happen is that it's going to release the four sporozoites. And these sporozoites, they are going to go and attach on the mucosa of the epithelial cells. And then they'll start releasing enzymes, and those enzymes, the same sporozoites, those enzymes they release, they'll start to break down mucus. And when they break down mucus, remember this place is covered by a layer of mucus. So they've broken down mucus, and then they'll be able to go and attach. It's like a ligand, it goes now and attaches to the receptor. So there is that receptor ligand interaction. And then what happens that now the sporozoites, when they have attached to the receptor of the apical of the epithelial cell, these guys, the the sporozoites are going to now start releasing their contents. They have got organelles on their apical end. They will start releasing those organelles. And what those organelles do is that they are going to cause the host membrane to form protrusions. So here is the guy, here is the crypto. It has released the organelles. And remember, it had attached here. So the host is now going to start forming these protrusions like this. And after it forms these protrusions, what will happen is that they will surround the sporozoites and then encapsulate it. It will reach like that, forming a capsule. But this capsule is not inside the plasma membrane of the cells, but it's just within these brush border cells. And what is going to happen after then is that this guy that has been formed is known as a parasitophorous vacuole. So abbreviate it as PV. Parasitophorous vacuole. So within this parasitophorous vacuole, the sporozoites which are there are going to start transforming. So the sporozoites are going to change into a trophozoite. The trophozoites are now going to convert into what is called a merozonte. Merozonte is also known as a schizonte. Please, it's not a merozoite. It's a merozonte. Hope will not confuse those. Okay. So we are saying it's this word merozonte. And merozonte is also known as a schizonte. So we have converted them into a merozonte. Now these merozontes are later on going to undergo a sexual division. Because these are merozones, we also call it merogony. That's asexual. And here you are going to form two types of merozones. You have got the type 1 merozones. Each type 1 merozone has got 8 merozoites. And then it can as well form the type 2 merozones, and each one of them has got 4 merozoites. What happens is that the type 2 is the one, so the type 2, if it is the type 2 that we formed, the type 2 is going to undergo gametogony. It is going to end up forming the male and the female. The male, males for the worms are usually small, so we call it a microgamont. It's this word, gamont. So we're going to form the microgamont and macrogamont. And what happens is that the microgamont, which is the male, is going to produce microgametocytes. You get that? The gametocytes. And then the gametocytes now, the microgametocytes are going to go and fuse with the macrogamont, which are the females. And fertilization is going to happen. We are going to form a zygote. When we form this zygote, the zygote can now transform into an oocyst. The oocyst can either have a thin wall or a thick wall. So if it is a thin walled oocyst, a thin wall is going to reinfect the host. Thin walls infects the host. But if the oocyst that we are forming is a thick walled, it is going to be released into feces and then it's going to go into the environment. That is the life cycle of Cryptosporidium parvum. So the way this guy is going to affect our body is that Actually, it can occupy any region from the colon, or it can colonize any part of the GIT from the pharynx to the rectum. That's about crypto. Besides that, it can cause the villi to atrophy. So atrophy of villi means that there's going to be malabsorption. So it causes malabsorption syndrome. That's another important thing we need to know about this guy. So that is that. So we've described it. So you can see the answer there is mature oocyst with four sporocysts. We've already disqualified the other option, isn't it? As zoonotic potential and infection being acquired by ingesting oocyst with two, not two sporozoids. That's supposed to be four. So we've already disqualified the other options. And then question 14. Which of the following has a correct life cycle of isospora berry? I remember we said that isospora berry is similar to Cryptosporidium parvam. For this guy, as we already mentioned, different from crypto where the habitant is like you starting the large intestine, the small intestine, the appendix, the respiratory system, the rectum. The habitant for isospora berry is basically the intestinal mucosa, and you are going to basically find it as intracellular. Specifically, it's going to be found intracellular in the jejunum or the duodenum. And the way the infection is transmitted is that you are going to have ingestion of oocysts. And remember, oocysts contain two sporocysts, not like crypto where I've got four sporozoids. And the infection is as well fecal oral. But how does it happen? So there's going to be number one, there's going to be ingestion of the oocyst, which contains the sporozoites. Now, again, I'm mentioning the word sporozoites. That is because, firstly, what happens is that actually the oocyst contains what is known as one sporoblast. This sporoblast is going to divide into two sporoblasts. The two sporoblasts are each going to differentiate into sporocysts, and then the sporocysts are going to form the sporozoites. So the mechanism, we are taking it from the sporozoites. So those sporozoites, we are ingesting an oocyst, and we are saying that the oocyst has got the sporozoites. And these sporozoites, what they will do is that when they enter, this is orophacal, they are going to invade the small intestine epithelium. Take note, there is invasion there. And when they do that, just like for the crypto, sporozoites convert into trophozoites. And then the trophozoites are going to convert just like crypto into schizons. The schizons, what do? They produce now merozoites. It's just that there we are type 1 and type 2. Okay. And then these merozoites will further differentiate into multinucleated merons. You get that? Merons, not merozoites. The merons are multinucleated. Okay, like that. So they can further differentiate. And then, just like we said for the other one, because for crypto had taken it, we had said the merozones were the same as the schizont. We are forming the merozones. It's not that different. It's basically the same. So when you have got the merons here, they can differentiate into the male and female microgametes. And those microgametes, the male and female, they can now come together to form a zygote. That zygote, so these guys, they form micro and macro gametes, not gametes. On top, we are talked about the gametes. So it will be the micro and macro gametes, and then so those gametes, I'll just say micro plus macro, they are coming and then they are forming a zygote. The zygote can form again the oocyst. The oocyst will go back. It will undergo sporulation to form two sporocysts. And remember, two sporocysts, each sporocyst has got four sporozoids. This one will form two sporocysts. Please take note, each one of the sporocysts has got four sporozoids. Okay. So where are you going to find these guys? So when you get fresh past stool, you are going to see just an oocyst, which has got one sporoblast. And then when you get a maturing oocyst, it is going to have two sporoblasts. Those are sporoblasts. Remember the fresh stool? The oocyst has got only one sporoblast, the maturing two sporoblasts. And then a mature oocyst is going to have two sporocysts, and each sporocyst contains four sporozoids. I think that's important to take note. And these guys, the way they are going to cause infection is that they're actually going to shorten the villi, and then they can as well cause the crypts to hypertrophy. And that is what causes problems. They can as well cause ulcers. So you can see why I picked this option. Ingestion of mature oocyst is going to form sporos, is going to produce sporozoids. Sporozoids transform into sporotrophozoids, which will form the merozoids, which are released in other cells to become trophozoid schizons. And then the schizons will form merozoids. I know it's a bit, not too good. The merozones again transform into gametocytes, micro and macro, and then you have got a zygote and oocyst. And if you can just follow this, let's follow it from. We have got a trophozoite there, trophozoite there. Of course, the schizont is what is going to produce the merozoite, right? So here the merozoites are released from each schizont. And then these merozoites, we said they are going to form the merons. They can as well take another form. And here we said they can as well form schizons, which will form other merozones. The merozoites, sorry, which are these guys here, which are released from the schizons, can transform into the gametocytes, which you can see, which can undergo fertilization to form the oocyst at the end. Ascaris lumbricoids infection can be associated with ingestion of infective larvae in food, ingestion of unfertilized egg in food, ingestion of infective embryonated egg in food and water, ingestion of larva in food. What is the way by which Ascaris is spread? What is the infective form? It's even there. And maybe in case you read something else. Okay. So as we said, remember when you're talking about the worm has got different phases. You have got L1, L2, L3. L1 is the rhabdite form phase. L3 is the filariform. Most of the worms, this is the infective form. And this, you are going to have the Strongyloistecularis here. You have got the Ancylostom ascaris. And you also have the hookworms, Ancylostoma duodenale and the necata. This is their infective form, L3. So ingestion of infective embryonated eggs in food and water. Yes, these embryonated eggs are the ones that actually will contain this larva. Remember, they're embryonated, not just any egg, but the embryonated egg. And we take them in the filariform. larva. It is an infective form of Strongylosterolis for Ascaris and the hookworms. Right. So embryonated eggs are the ones which contain this gamma larva. Remember, embryonated. The eggs already have the embryo, and the embryo is that gamma larva which is there. And then number 16. The following applies to Tinea solium except cystosecai celluli in pigs. This is one thing we should know. This is true or false? Let's start A. Is it true? This is true, thank you. This guy will be even causing cysticercosis. Strobilus consists of immature, mature, and gravid proglottids. True or false? This is also true. They are hermaphrodites. True or false? True. Single proglottids can contain up to 200,000 eggs. True or false? So this is false. What are hermaphrodites? Anyone that wants to help there? Thank you so much. Those guys which have got both male and female in the same womb. So these are the guys. Number 17. This is true about helminths. They are the answer is D. Each proglottid contains 40,000 eggs. Yeah, 40,000. This is true about helminths. They are unicellular. Now helminths are not, right? They are they have got a lot of cells. They actually multicellular, spend little time in human host. Hmm. They spend time there. Some worms we can actually be with them for life. Divided into three groups. No, how many groups are? Oh, actually, that's the next option. They are divided into two. Which two groups are helminths divided into two? Thank you. The nematodes and the platyhelminths, or flatworms and roundworms. The following pairs are correct regarding nematodes. Remember what we said, the simple mnemonic, if you want to remember everything. We said oviparous. You can use the common mnemonic. Tana. Do we have Tana in class? You can be talking about a person. And then you have got the viviparous, which are the rest. And then you have got ovo, viviparous. And then here you only have one strongyloid. So if it is not trichuris, it's not ascaris, it is not ancylostoma, it is not necata, and it is not strongyloid, then it is viviparous. That's the only thing we can know. So you can see the only way you can be viviparous is if you are not Turner and strong. So we can actually make it like this Saturn, right? Saturn. Yes. This is strongyloid, ancylostoma, trichuris, and then ascaris, necata. If you're not Saturn, then you are viviparous. Ish, this mnemonic. No. Okay, number 19. Ocular cysticercosis can be diagnosed by. We know cysticercosis. Let's mention about it back. When you're talking about tenure, we have got two guys which we call tenures. We have got sodium, which is a good guy. Sodium. They are not good guys, they're bad. And then we have got Saginata. Now, the sodium guy, you can have adult worms getting to infect us, or you can have the larva. Infection caused by the adult. What kind of infection is that? Teniasis. And then by the larva is basically the baby boy, right? Yes. What does a baby boy cause? Yes, cysticercosis. Imagine it was writing this word cysticercosis. I failed to write it in that quiz. Adults, these guys cause teniasis. So, because saginata, this camalava doesn't affect human being for saginata. There is no cysticercosis. That is an interesting thing that we should be able to know. I'm forced to mention one thing. When you're talking about tinea, sodium, we called this guy as an armed tapeworm, also known as pork tapeworm. Saginata is also known as unarmed, also known as beef tapeworm. Saginata is like, it goes up to 15 meters, very long. The average for sodium is like 5 to 7 meters. We are talking meters here. This is a worm in the body of a human being, 15 meters. So, meaning that a human being can go there at least somewhere 7 times if you are 2 meters. Okay. And another thing we need to know about the adult, or I can just say tinea, is that it has got four suckers plus 25 to 50 hooklets. So it has got suckers and hookers. Remember we said cystodes are going to have suckers and hookers. Trematodes. The only trematode that is more important is cystoma. Schistosomiasis, that one which causes is that lymph schistosomiasis, bilharzia. So it has got 25 to 50 hookers. Please take note of that. That's important. And then 40,000 eggs. I think that is that. About cysticercosis, how is cysticercosis caused? You need to know this, please. Cysticercosis number one, it can be caused by ingestion of eggs. Please, teniasis is caused when you chew the muscle, you chew the same beef, or you chew the pork, that the muscles that contains this guy, the tinea. So besides the eggs, for you to get cysticercosis, you can as well get it in what is known as external infection, autoinfection, and internal. And we already did explain this. The internal is where you have got the proglottid going back to the stomach in reverse peristalsis, and then the external is this guy is going to penetrate through the large intestines and then it goes back into blood circulation. Okay. So for the cysticercosis, it can affect a lot of regions. Actually, what you are going to see is that most of the regions that it affects, there will be nodules, like these elevations on the skin. It can be on the tongue. It will also go in the brain where to form these masses in the brain, on muscles, skin, like you're going to see these masses. It can as well cause ocular cysticercosis in the eye, affecting the anterior chamber. And we are going to see a grayish mass, which is whitish, in the anterior chamber. And actually, when you see the scolex, remember the scolex is the head of these guys, the nematodes. When you see the scolex, that is like a definitive diagnosis for ocular cysticercosis. That's the meaning of pathognomonic. It is a definitive diagnosis when you see the scolex. So whitish grayish mass in the anterior chamber of the eye. And the scolex is pathognomonic. Regarding Prasquanto, it is It's given as a 3 to 4 day course, 40 milligrams per kg for it to work well. Its efficacy is restricted to Cystosoma mansoni. It is given as a single dose, A and C. So I see some guy, I don't know who answered that paper, picked A and C. Okay, so the thing is, yes, Prasquanto is given as a single dose. It is given 3 days as the 3 day course. But this, you don't give it as 40 milligrams per kg. So specifically the lecture notes from the lecturer who taught says 10 milligrams per kg. From the trusted sources, I was trying to find out the WHO recommended dosage for Prasquanto is 20 milligrams per kg. So this just disqualifies this option. It is given as 10 milligrams per kg according to the lecture notes, so 20 milligrams per kg according to the research I made for 3 days. So that's why the A and C I removed it and picked just C. The habitat is the small intestine. Now remember we said if something is not trichuris and enterobius vermicularis, then it is small intestine. These are the only two guys that stay in the large intestines, right? So the habitat is a small intestine. Ascaris, yes. And then Necata, yes, because it's not Tinea, it's not Trichuris, and it's not Interobius. So this is the only one that's out. So the answer is A and B. Trichomonas vaginalis can be diagnosed by. Now, as we said, Trichomonas vaginalis, we say, let me freshen up our minds. What's the infective form of Trichomonas vaginalis? I- The trophozoite. Thank you so much. And because this kamagai is causing, infection of the female reproductive system, basically the reproductive system and the urinary tract, you are going to get that kamahai vaginal swab. And when you get that kamahai vaginal swab, the pap smear, you are going to go and look at the cells. Okay. In the diagnosis, of course, you use the pap smear. It's not the past cells really. You are looking at the trophozoite. So the best option there is B, finding trophozoites in high vaginal swab. Pap smear is also used. Pap smear, you look at the cells. Past cells, these are white blood cells, and they're not the ones that we look at. 23. Anemia can result from infection by the following. Oh, this one should use water. I think so, but trichuris, especially in heavy infection, can lead to a lot of loss of blood when someone goes to the toilet. There is much loss, especially with heavy infection. So it can result in anemia. And besides, these guys also, imagine it has affected, because this guy affects, it resides in the large intestines. In the large intestines. Now, what I wanted to mention. Yes, Doc, want to say something? So the high vaginal swab is going to show the trophozoites. Like when you take a high vaginal swab, you'll be able to see the trophozoites. For the pap smear, remember, pap smear will get the cells, right? No, the past cells are white blood cells, and that's not what we are trying to look at. We're not trying to look at the white blood cells. Hope that's okay. From the lecture notes, you are going to find pap smear is one of the diagnoses, but it doesn't look at the past cells. Okay, number 24. Cystodes are characterized by the following except. Remember cystodes, sexes are separate? No, these guys are monoecious, right? Monoecious. They have got the same guy. Let's just say platyhelminths the same guy is female and male. They are hermaphrodites. Hermaphrodite is the same as monoecious. This means that same parasite with male and female. That's true. Oviparous. Remember we said cystals and trematodes are both oviparous. They all lay eggs. Have suckers and some have ookas. This is true. Have suckers, some have got ookas. But for the trematodes, what if this statement was talking about trematodes? How would we put it? They have suckers but no hookers. Thank you so much. Number 25. Yes, please. Thank you so much. That's a very important point that the doc has mentioned there. Of the flukes, you have got only one guy which causes bilharzia cystosoma, and that guy has got separate sexes. The rest of the platyhelminths have got the same helminth being male and female. Only cystosoma, which is a fluke, it is a trematode, it is a platyhelminth, it is a helminth, it is a parasite. Hope that doesn't confuse someone. Regarding Isospora bari, we did mention something about this guy. Infection is acquired by ingesting mature oocysts with two sporoblasts, with each containing four sporozoites. Why is this option not correct? Pardon? So, what is this supposed to be? Thank you so much. So, it's supposed to be two sporocysts, each containing four sporozoites. And then B, infection is acquired by ingesting mature oocysts with two sporocysts, each containing four sporozoites. Yes. So, that's the answer. The rest will be wrong, right? Because it's a multiple choice. 26. The ideal drug for use in intermittent presumptive treatment of malaria in Zambia is what? Pyrimethamine, coartem, sulfadoxide, and then we have got pyrimethamine and sulfadoxide. So let me mention something about these guys. Please don't forget the incubation period for Plasmodium. Remember, as we've been saying, normally the ovale and vivax move together in most of the things. So vivax and ovale, their period, their incubation period is on average 14 days, but that is from it can either be from day 8 up to day 31, any day between here, average being 14. And then you have got another guy, the waste guy, Falciparum. Falciparum incubation period is 12 days. That is like from 8 to 14. It has got like the shortest incubation period. The longest incubation period is for malaria. That's 28 days. And that ranges from, it can be from 8 days up to 40 days. So let's take note of this. Another thing that I should again mention is that ovale and vivax can cause relapse. Relapse means that they form hypnozoites. Hypnozoites hide in the liver and they can come and cause infection later on. But these two, falciparum and malaria, they are responsible for recrudescence. And recrudescence is where, let's say you are treating, but you have not managed to wipe away all the parasites. So those few that have remained later on, they can arise and cause infection. Another thing that I want to mention, because I saw a comma question, is that when you're talking about how these guys cause infection, the malaria guys, they have got proteins that help them to adhere to surfaces. When you have got the parasite, it will be adhering to surfaces and that helps it to cause infection. For example, you have got proteins like what is known as the Plasmodium falciparum endothelial membrane protein 1. So you have got this and then you have got also HRP2. What's this? Is this histamine release protein 2? Yes. So these guys will be able to bind to ligands on the endothelium. So the parasites will be binding to the endothelium. And the thing is that actually when one plasmodium binds to the endothelium, just like the way platelets behave, they can as well bind to other plasmodium parasites. So they'll be like clamping together like that. So these are proteins that are produced or found on the plasmodium. And these will be able to bind. You have got ligands. You know a ligand is like an hormone. It has got where it binds. So you have got ligands that help in the binding of these plasmodium guys to the endothelium. Like CD36, you have got VCAM1, and then you have got ICAM2, and then you also have E-selectins. So these are ligands that will help for the malaria to be able to bind. Now, when it comes to the treatment, and I think this is important, you divide the treatment for malaria into different. Of course, we've done the pharmacology much in detail, but the microbiology is very short and concise that we need to know. Number one, about treatment, you need to know that the first line treatment for this guy is coartem. And you know coartem is a combination of artemisinin and the lumefantrine. Artemisinin plus lumefantrine. The second line that we can give for the treatment of malaria is quinine, and we'll give it orally. So oral quinine. And then when someone has got severe cases, when we say severe cases, we can as well say complicated cases. And you know complicated cases are basically caused by what? The falciparum. So for the severe cases, you can as well give attenuate. Specifically that one you are going to give it by injecting. Injection can either be IV or IM, isn't it? So attenuate. And then that is going to be the first line. You can as well give artemether IV, or I mean IM. That's for severe cases. Artemether. You can see also the another A there. And this is IM. You can as well give quinine. Quinine can either be given IV or IM. And you are going to combine it with either 5% or 10% of dextrose. This is the complicated case which is caused by the falciparum. And then the uncomplicated cases, which are non-severe, these guys, the first line of treatment is going to be quinine. And quinine can be given in all trimesters, first trimester, second, third. It's not having any effect on pregnancy. Another one that you can give for uncomplicated cases is attenuate. Specifically, you are going to inject it. But this one only give in the third and in the second and third trimester. Don't give it in the first trimester because that's the point where having development can interfere with development. You can as well give Coartem. For Coartem, you can give it also in the first trimester, so it is okay in the first trimester. Remember, Coartem is a combination of Artemether and Lume. What's that word again? Lumphe, Lumenfantrine. And then the last one, that's for the complicated and complicated. And the last one is the intermittent preventive treatment. I can put it as IPT. Intermittent preventive treatment. For this one, what you use is a combination with these folic acid antagonists like the sulfur, doxamine, and pyrimethamine. Are these the ones? We know the folic acid antagonist is supposed to be trimethoprim and sulfur methoxazole. These are the ones that you give for the intermittent preventive treatment. This question is saying the ideal drug to use for intermittent presumptive treatment is, so we've picked that because that's the one. Preventive, presumptive. No, presumptive, this is presuming you are doing it in advance. I don't know if that was a hand or something. 27, Tenia sodium belongs to the class. We know that this is a cystode, isn't it? Second, the what has been given here, Platyhelminth, Tenia, Animalia. Animalia is a kingdom. Platyhelminth is the phylum, just like we also have Nematoda. And then the class is Cystoda, the order their family is Ctenidae. So this is a family, genus Tenia. Yes, please. Is that a hand? Okay. Which of the following has the correct morphological life stages of Cryptosporidium parva? Now, already did describe the life cycle of crypto, isn't it? Mature oocyst is taken, it causes a release, it releases the sporozoites. Sporozoites is going to also release, it's going to convert into trophozoites. Trophozoites, you have got type 1 merons, which remember the type 1 merons? These ones will remain and infect. They can convert into merozoites and then infect the cells. Or the merozoite can transform into the type 2 merons. And the type 2 merons can further convert it to merozoites. And these merozoites can form or differentiate into gammons, the micro and macro. Remember we said that the micro is gammon is going to differentiate into micro gametocyte, which will join with the macro gammon to undergo fertilization to form a zygote, and then the oocyst. We are going to form a thick or thin walled oocyst. We said that the thick one is going to be released in feces, while the thin can cause auto-infection. If you look at option 1, mature oocyst, trophozoite. After trophozoite, actually trophozoite is not what comes after from the oocyst. It's supposed to be the sporozoite. So that's not correct. And then option C, mature oocyst zygont is not zygont first. Mature oocyst is sporozoite. From sporozoite, schizont, and then schizont, trophozoite. You can as well see trophozoite is supposed to come before schizont. because the schizont is a merozont, which is going to form the merozoids. The following can be transmitted by mosquitoes. Mosquitoes, I wonder why these questions about virology were in parasitology test. That's why. So mosquitoes, these guys are arthropods. And for viruses, you have got those viruses which are spread by arthropods. We call them arboviruses. Viruses like chikungunya, dengue, West Nile, right? Those are arboviruses. They are spread by arthropods like mosquito. Taenia solium rosterum possesses about, we already did mention this, 25 to 50 hooklets. Remember what we said? These are the hooklets for Taenia solium. During sporogony in mosquitoes, gametocytes are set free in the hindgut. Sporogony in mosquitoes. Let's describe anyone that can help with a small description of the life cycle of a mosquito, like what's happening in the mosquito life cycle. Anyone? Are people around? Okay, let's do this. Remember, i- in a human has been bitten by a mosquito, sporozoids have been released. Sporozoids. When these guys go to the liver, they are going to change into what? We can take it like this so that we can discuss together. What do they change into when they go to the liver? Anyone? I didn't get that. Would you say that again? They are going to convert into they are going to convert into a merozoid, isn't it? Okay. And you know these merozoids, what happens is that the doc that said schizont, that's not false because merozoids actually stored, like they are hiding in the schizont. So these merozoids are going to be released when the hepatocytes rupture and then they are going to enter blood. In blood, what happens? They are going to transform and they are going to form again schizons. Actually, what is going to be produced when the liver ruptures is going to be the trophozoites, isn't it? So those trophozoites are going to be released and then they are going to go to the red blood cells. In the red blood cells, they are going to again start another cycle. So we are saying the merozoites here, which are in the liver, they've been released when the hepatocytes gets to rupture. So sporozoids first have entered the human, they go to the liver, and while in the liver, they are converted into a schizont, and each schizont contains a lot of merozoids. When the liver ruptures, there is a release of the merozoids, and it's actually the release of these merozoids that causes the fever. When the merozoids are released, they are going to change and form trophozoites. When they form these trophozoites, they are again going to form a schizont. Remember earlier on, the merozoites were also released from a schizont. These schizont, later they are going to form again merozoites. These merozoites can as well infect other red blood cells, and it continues like that. If this was vivax or ovale, the merozoites can as well, which are in the liver, can end up forming hypnozoites. What happens in the mosquito is that a mosquito has come and gotten a blood meal, it has now been infected. So in the midgut of the mosquito, what happens is that because just these guys that we had produced here, the merozoites, can undergo gametogony, where they are going to be forming gametocytes. The gametocytes which are going to be used to make others. So the merozoites are going to be able to pass through the trophozoite, schizont, merozoite. They will cause again the rupture of the red blood cells, but they can be converted into micro and macro gametocytes. Those gametocytes, when they go into the mosquito, when it gets a blood meal, specifically in the mid-gut of the mosquito, that will be the mosquito life cycle, they are going to now change, they are going to undergo fertilization. Remember, you have got both the male and female. They will undergo fertilization, and in fertilization you are going, please take note that one microgamete has got, actually not microgamete but microgametocyte, because those are the first ones that we have, this name gametocytes. For the male, which is micro, it has got 4 to 8 microgametes. The female, which is my macro gametocyte, only convert into one macro gamete. So one of the males will fertilize the female, and then when that fertilization happens, you are going to form the zygote. This zygote is further going to transform into what is known as an ookinete. The ookinete can further differentiate into an oocyst. This oocyst stores a lot of sporozoites. So what it will do is that it will now go to the mouth, salivary gland of the mosquito, and rupture to release the sporozoite. So sporozoites are now in the salivary gland of the mosquito. If it bites another human being, they are released into the human. So during sporogony in mosquito, gametocytes are set free in midgut and undergo further development. It's midgut. That's an important word here that I'm mentioning. Number 32. The pathogenesis of Cryptosporidium parvum in the human can be associated with alteration of osmotic pressure leading to chronic diarrhea, alteration of osmotic pressure leading to acute diarrhea, atrophy of intestinal villi does not alter the upper uptake of fluid, electrolyte, nutrients. Malabsorption syndrome can arise when there is alteration of osmotic pressure. So this is an important concept of crypto. There is malabsorption. I think we did mention this. There is malabsorption in cryptosporidiosis, because what happens is that when you have got, remember the stages that we said, you have formed your zygote, you have got the thick word oocyst, the thin word, the thick goes in feces, the thin word. So the crypto is going to go and attach on the surface of the intestinal mucosa, and there it is going to cause a profuse watery diarrhea. Take note of the watery diarrhea, and then weight loss is going to be happening. Because we said that this guy, crypto, remember, it occupies any part from the pharynx to the rectum. It as well causes the atrophy of the villus. Because of that atrophy of the villus, it also causes malabsorption. So that is what happens. It causes a profuse watery diarrhea, not these ways they are saying. And of course, yeah, it is that is going to be an osmotic diarrhea. Okay, thank you so much for that. Anyone that has a thought? What is wrong with option B? So anyone that can help before I say anything that I'm thinking? All right, thank you. So would we say A is correct in such a case? So the only yes, doc, please go down. Yes. Mm, so why is B being disqualified? Okay. So from what you've read, what have you read again? Oh, thank you so much. So yeah, that is sensible, right? That is so sensible. I didn't even take time to look at this. I thought there is a word and somewhere. So malabsorption syndrome can arise when there is alteration. Malabsorption is the atrophy, yes, the atrophy of the villus. So I thought these were two different options which have been combined like there is malabsorption and alteration. So this is not there. Yes, this is the best. So there is at the alteration of osmotic pressure. My absorption is about atrophy of the villi. Thank you so much for that. The correct life cycle of Tinea sodium parasite is characterized by, didn't we already describe this? Tinea sodium, man eating undercooked pork, man with tapeworm in the small intestine, and then you have got the gravid proglottids passed in feces, and then the pig will be able to ingest those, and then oncosphere penetrate the small intestine, that's obviously of the pig, and then they are going to go to blood. From the blood, there's going to be filtering in different organs, various tissues, and there they will cause a cysticercosis. If you look at a man eating undercooked pork, man with tapeworm, it's not large intestine, that's why that one is out. Even this one will be out because of large intestine. And then the man eating undercooked pork, man with tapeworm in the small intestine, gravid proglottid past in feces, pig ingests eggs, oncosphere penetrate the large, again there's large intestine there. So that's not true. Penetration is happening in the small intestines. Which nematode is not a soil transmitted helminth? Enterobius vermicularis, Ascaris lumbricoides, Strongyloid stacularis, Trichuris, trichuria. Which ones are the other soil transmitted? The dog gave a nice mnemonic to be able to know the soil transmitted helmets. Anyone who remembers the mnemonic that was given? NASA. Okay. NASAT. So you have got Neketa, Ascaris, Strongyloids, Ancylostoma, and you have got the Trichuris. I think I'd combined two of these to make H because those are hookworms. So you can just say, oh, this is what I was saying, Nata. So you just put H with here, but there is no another A. Anyway, the NASAT is just fine there that we can know. So Enterobius, you can see it's not part of those, so it's not. How is Enterobius spread? Anyone that can help with that? Thank you so much. So that one is fecal oral. The gold standard for the diagnosis of malaria. So malaria, the gold standard is microscopy. So you are going to use the James 10 thick and thin blood smear. That's a conventional method. Which of the following statements is correct about Ascaris lumbricoids? Associated with clubbing? So this one is not. There is a specific one that is associated with clubbing. Anyone that knows the specific guy associated with finger clubbing? Yes, trichosis. And then B, infection transmitted through skin penetration. Again, we have said this guy, you have got the eggs that are going to be taken, isn't it? The Ascaris. Heavy infection of adult worm inhabiting the large intestine can cause intestinal obstruction. Remember, for large intestines, we only have two guys. Which ones are those? So, trichurias and enterobius. Right. Heavy infection adult worms inhabiting small intestines can cause intestinal obstruction. So, this intestinal obstruction that is caused by Ascaris will actually lead to deficiency of some nutrients because there's not going to be any absorption because of the obstruction. Right. Teniasis is transmitted by ingestion of eggs in food and water, consumption of undercooked or raw pork infected with mature cyst, external autoinfection, internal autoinfection. Why do you think the other options are wrong? Anyone that with any thought? Yes. Thank you so much, Doc. Yeah, that's the basic thing. I've even put it down here. Tiniasis, that's where we get the undercooked or raw pork. For cysticercosis, that's where you can inject the egg, or external to infection, internal to infection. We already described what these are. I want to mention again something on Ascaris lumbricoids. Ascaris lumbricoids, the worms don't attach to the mucosa of the small intestines. But the way they'll be working is that they're going to be shortening the villi. That leads to the obstruction of the, because they'll be accumulating, there'll be obstruction of the small intestines. And actually for the Ascaris, it can migrate to the liver for the male worm. It can migrate to the liver, and there it can cause obstructive jaundice. Also take note that Ascaris is also associated with lactose intolerance, vitamin A malabsorption, because it also undergoes the lung heart migration. It as well causes Lofra syndrome. So in case you want to help your water, don't use chlorine, use iodine. Another thing is that because these guys are spread in the soil, of course there are a lot that are spread in the soil that we have talked about, but specifically for an Ascaris, you know, there is this word pica. Pica is just that pulling force to eat non-food substances like soil. It happens mostly in the pregnant, those who are, women, and they have got higher chances of getting ascaris because of loving to eat soil. Even children, because we also used to eat soil when we were children. 38. One of the most important or primary vectors for malaria in Zambia is Anopheles aegypti, Anopheles funestus, Anopheles costan. So the funestus is important for malaria in Zambia. It's like the most important, one of the most important. Funestus. The correct life cycle of Plasmodium falciparum in both the intermediate, yes, doc, or the filarial worms, yes. All right. And then the correct order of life cycle of malaria in both the definitive. The definitive host is the mosquito, intermediate host is the man. So in the mosquito, there's going to be ingestion of the micro and macro gametes by mosquito during blood meal, and those are going to differentiate in, these are micro gametocytes. They will differentiate into microgametes. I've now seen this D, which I've picked. This is not saying microgametocytes. Okay, I'm going to go back to A and check it. And then we are going, so let's check A. It's supposed to be microgametocytes first. Ingestion of gametocytes by mosquito during a blood meal. Differentiate into micro and macro gametes, zygote, ookinete, oocyst, sporozoids. Definitive host sporozoid in sporozoid injected by mosquito during blood meal, schizont in liver, mature and release merozoites into blood circulation, invade the red blood cells, young and mature trophozoites, schizont transform into merozoites, infect other red blood cells or transform into gametes. Okay, this is making much sense. Let's see B. Ingestion of gametocytes by mosquito during a blood meal differentiate into micro zygote oocystokinase sporozoite and then definitive sporozoites ingested by mosquito during a blood meal schizont in the liver and mature releasing merozoites into blood circulation invade red blood cells. Schizont transform into trophozoite, merozoite, trophozoite transform into merozoites, and then merozoites infect other red blood cells or transform. Are these options even different? I'm not even seeing the difference. The hosts. So for B, like for B, what's the difference between B and A? Okay. The intermediate the intermediate host doesn't ingest gametocytes. Okay, so there's a difference. This is ingesting, ingestion. No, but that's still not making sense really, because it's the mosquito getting, isn't it? The mosquito is getting the gametocytes. Or it's a definitive yes. Oh, they've swapped these things. Okay, these words have been swapped. And then C, definitive host, which is the mosquito, ingestion of micro, so this is out, micro and the macro gametes by mosquito during blood meal, differentiate into micro and the micro gametes. Again, this is gametes to gametes, zygote, and then the ookinete is also not true there. So that still leaves. Again, A is intermediate host, which is so this one is also not true because it's the intermediate host ingesting the gametocyte. So that just remains with D, ingestion of micro or macro. But these were not supposed to be gametes. Again, they differentiate into gametes. I feel these were supposed to be gametocytes and then differentiating into gametes. Otherwise, it's a correct one. Or maybe that's what you read, it's gamete and then it differentiates into the gametes again. Anyone that can help there? Anyone that can say something? No one. Okay, this is an error. This is supposed to be macro and micro gametocytes. They are the ones that will change into the gametes. But it's the best option that we got. So number 40, the most commonly involved area in amoebiasis is ileum, terminal portion, cecum, liver, rectosigmoid. Does someone have a different answer here? So in amoebiasis, what is infected normally is the terminal ileum and the cecum. These are the portions that are infected because the infective form, that's the cyst, isn't it? Because the trophozoite doesn't really stay it when you take it by mouth because the infection is orophycal. By the time it will be reaching the stomach, it would have been destroyed because the trophozoite can't stay in the acid. So the trophozoite is just used for growth. It is the growing form. What else should we know there? Another thing, the trophozoite has got pseudopodia. This with my hand it will be used for moving. And then it is going to divide by binary fission and budding. The cyst is going to be seen in unfavorable condition because it can withstand those conditions. So when there are unfavorable conditions, there is what is known as incystization. Incystization is the formation of a cyst. Conditions become favorable, you now form back your trophozoite, and that's known as excystization. Existing this guy. Another thing we should know is that what else? Basically, this guy is affecting the two ends that we've listed, but specifically, you are going to form like the when you're undergoing existization, where you are releasing the trophozoite. The trophozoite has got 8 nuclei. First, it starts as four. You have got four nuclei, so it's known as a quadrinucleate amoeba, also known as a metacyst. So when first the cyst is ingested, it's going to go to the terminal ileum. It undergoes excitation. There, it is going to release a trophos a trophozoite or the amoeba that has got four nuclei, that is known as a quadrinucleate amoeba, also known as a metacyst. And what happens is that because there are four nuclei, they will undergo cell division, and you are going to end up with 8 nuclei. And then each one of the nuclei is going to end up being surrounded by a cytoplasm. So you are going to form now what is known as amobule, also known as metacyclic trophozoite. And that one is the one that is going to colonize the submucosa of specifically the cecum and the large intestines, and that's how you end up with the amoebiasis. And there we're going to be able to form a flask-shaped ulcer and also a granuloma like structure, which is known as an amoeboma. But this happens a bit quick. Okay, so cecum there. Which statement is not correct about Ascaris lumbricoides? Parasite is associated with ascarine pneumatitis. This is true, right? This guy, because it undergoes blood-lung migration, it can cause inflammation that side. So Ascaris pneumonitis. High prevalence of infection is due to the ability of eggs to survive under a variety of environmental conditions, and that is true. They are oviparous. Remember we said the oviparous? Again, what is the common mnemonic for oviparous? Saturn. Actually, just sana. This T will tell us everything that is not uh, viviparous. So Saturn is for everything that is not viviparous, but if we remove the T, it's going to be for those which are specifically what? Actually, not even the T here. Yes. So SANA, Sana or NASA, these guys are the ones causing which are oviparous. First passage through the liver is associated with remarkable pathogenic changes. That's not true. Of course, the chamomile can go to the liver and cause jaundice, but it's not remarkable pathogenic changes. 42. The following symptoms pertain to tinea solium. Okay, tinea solium infection. Specifically, these things mentioned here is in neurocysticercosis. We just need to know things that when the brain is affected, what happens? There's a disoriented behavior, transit paralysis, where there's paralysis that is happening. Disequilibrium, you can think of the brain is affected, equilibrium is affected, meningoencephalitis, failing vision, and also a lot of eosinophils in the central nervous system. Look at A, disequilibrium, iatad cyst is what disqualify that, and then B, what is disqualifying is swimmer's itch. Which one causes the swimmer's itch? Yes, please. the cystosoma. Thank you so much. And remember, we've got a lot of those: hematobium, Manzoni, disorder, and then this other one. What is disqualifying it is the pulmonary, and the neuro. Okay, the following can be isolated in urine specimen: cystosoma interlatum. As we have said, this guy is the one which causes urinary bilharzia or schistosomiasis. Wuncherella, cystosoma mansoni. So the rest, no. But we can think of Wuncherella causes. Which, yes, please. I'm hearing some moving hands. I don't know if those are contributions. Is that a contribution? All right. So I was saying, what does the cystosoma mansoni cause? First, Wuncherella bancrofti. So that is okay. Wuncherella. This is a lymphatic filarial worm, right? So it will cause a lymphatitis. And then cystosoma mansoni. Anyone? So this guy causes bilharzia. Number 44. The following has the capacity to produce group infection. Okay, group infection. Just think of vermicularis. Vermicularis where children are normally scratching their anal area, and as children, unless maybe we've as when we were growing up, they'll just remove your young brother, the short, and they put you in. And remember, my other brother was scratching, so the worms remained there, can spread around like that. So that's Enterobius vermicularis. 45, regarding antihelminth use in. Yes, please. Cystoma intercalatum, one that causes urinary bilharzia. The following has a capacity to produce group infection. We already answered that. Yes, please. Yes. Oh, thank you so much. So a correction there, we are going to find it. So if it passes in stool, how are we going to use urine as a specimen? Unless we say it's similar. So if it is similar, then it is as well going to, since it's similar to hematobium, it might as well lead to the same, getting the same diagnosis of the urine. Bancroft in urine, elephantiasis, that's supposed to be blood. Okay. So would go for Bancroft. Okay. Thank you so much. So the doctor said, would you say those again? Cystoma? Which cystoma did you mention? Okay. So cystoma, hematobium, trichomonas, vaginalis, and Wunschelera. You can get them through urine. Trichomonas causing UTI. But I need to revisit my source if it lied to me. Okay. So intercalatum is out there. We are saying it is Wunschelera bancroft. I'm still going to come back. I just double check my source because it was showing me it causes urinary amoebiasia. The following has the capacity to produce group infection. This is the same question. Regarding antihelminths use in pregnancy, which drug is ideal use stating starting with the first trimester? So from what I was trying to read from the lecture notes and other things, all these they've mentioned them. There is no known effect in pregnancy, including parental, palmoti, albendazole, mebendazole. They don't know the effect. But I think we can remember we said if you want this, you want to do the risk benefit ratio. So the closest that I came to was saying parental palmoti can be used, no restriction. Then it was saying albendazole is a group C drug, and you know group C, this means these guys have got restriction to be used in pregnancy. The pathogenesis of Ascaris lumbricoides can result from skin penetration, migration of larvae in the liver, lungs, and adult worms in the large intestines, migration of larvae in the liver, lungs, and adult worms in the small intestines. So this is true, right? Ascaris lumbricoides, what happens for this guy is that when we take it, because we are going to take it orophaco, isn't it, orally? So we've gotten it, it is going to go to the small intestines. But before it finishes, from the small intestine, it's going to penetrate and it go into blood. When it goes into blood, it's going to go to the heart. It's going to go, actually, it's going to also pass to the liver. It's going to go to the heart. It is going to go to the respiratory system using the capillaries. Remember, it has gone to the heart, so it goes via the pulmonary arteries to the lungs, and then they to pass into the alveolar. So when it is passing through the alveolar, there it can cause some little infection, and then it will go up the bronchial tree, and then it will be you are going to cough it up. It will now come back into the esophagus and then come back into the GIT, back to the small intestines, and that's when now it causes the infection. But it came from the small intestine. Why didn't you just go direct? But that is how it moves. Number 47. Which of the following statements is the correct life cycle of Gardia lambria? We've already described the life. Did we describe the life cycle of Gardia? Or that was Trichomonas? So for Gardia lambia, this guy is also going to cause these, STI. It's sexually transmitted. And where do you find it? You can find it in the intestines. Actually, when you're it's a flagellate. So flagellates can be classified as those which are intestinal or urogenital and those which are blood or tissue. Those which are found in the intestine or the urogenital, you have got Giardia and Trichomonas. And then those in blood, you have got Trypanosoma and Leishmania. For Gandia specifically, to it stays in the upper part of the duodenum. Such, so the first part of the nose we are saying upper part of the duodenum, the second part of the nose we are saying upper part of the jejunum and the duodenum. So anyway, take note that it's upper part of the jejunum and the duodenum, because at least like that, upper part of the jejunum is near to the duodenum. So that's where this guy is found. And take note that it is pear shaped and the structure is like this, something like this. So this is the back, the dorsal aspect. You can see this, it is convex at the back and then anteriorly. And when you look at it from like the front, it has got like this from a big eyes, something like this. And then it's going to have an exoster on the middle. That's Gardia. So this guy again, what you need to know is that it has got a cyst. The cyst can either be mature or immature, but the mature one has got 4 nuclei and it's actually the infective form. This guy is a spread fecal oral. Did I say STI? It is spread fecal oral, but it can of course be transmitted by these weird sexual practices. And remember, it's spread by fecal oral, but let's say for example these homo. What do you call these males who do that sin? What's that? Homosexual males. Because they are males, it means that one person is going to be using the penis in the mouth of the other one. And because of that, something that is supposed to be spread oral fecal is being spread by sex. You hope you get the sense. Otherwise, it's not supposed to be spread by sexual contact because it's oral fecal. Just a moment. Hope we can see. Are we? Are we able to see? Can someone confirm, please? Okay. So I was saying this Gardia is a spread or a fecal, but for homosexuals it can be spread by sexual contact. What else should we know about this guy? It is as well going to, because it is orophacal, it as well causes diarrhea. It's believed to be zoonotically transmitted from animals. Specifically, it is causing osmotic diarrhea. And these trophozoids, they can as well go to the gallbladder, and there they can cause jaundice. So that's a basic thing. So the patho- so which of the following statements is the correct life cycle of Giardia? Ingestion of mature cyst. Remember we said there is a mature cyst with 4 nuclei, and then it is in the stomach, and then the duodenum where it is going to form two trophozoites, okay, which are going to hatch from it from one cyst. Trophozoites are going to multiply by binary fission, and then they'll colonize the duodenum and the upper part of the jejunum. The trophozoites are going to adhere to the enterocytes, and then they are again going to incystize, they are going to form the cyst down the colon. Those cysts are now going to go in the feces. Yes, please. I've heard a hand. So option A is not correct because it cyst with 2 nuclei. No. Option C, mature cyst with 4 nuclei, existation in the stomach and duodenum. Two trophozoid hatch from one cyst. Trophozoid multiply by binary fission, colonize duodenum and upper duodenum. Trophozoid adhere to enterocyte, incystation down the small intestine environment, cyst passed in feces into the environment. Okay, what's the difference with B? Anyone that has noticed any difference with B? Ingestion of mature cyst with 4 nuclei, existation in stomach and duodenum. Two trophozoites hatch from one cyst. Trophozoite multiply by binary fission, colonize duodenum and upper part of the jejunum. Trophozoid adheres to enterocytes, excitation down the, okay, I've seen now. So B saying excitation down the colon. So the formation of the cyst is going to form as you go down the colon. Then this one is saying excitation down the small intestines. And then the cyst is passed into, it's saying the colon where I've got the feces, isn't it? That's where it's going to be passed into the feces to the environment. 48. Flea, fleas are vectors for organisms causing the following. So, bubonic plague, this is an example of what is known as the plague. There are three forms. You have got the pulmonic, this bubonic. The plague is actually called the V and not just plague. It's bad. Try to look at the history of the plague. It's caused by a senior pestis. So It's believed to be carried by rodents, and then we have got these fleas that can come from those same rats. They are not actually supposed to be like specific fleas, rat fleas, which are going to be able to get a Yersinia and then be able to spread it. Following symptoms may be seen in ascaris due to adult worms. Vitamin A malabsorption, as we said, ascaris, vitamin A malabsorption, intestinal obstruction, restless sleepiness, cholecystitis, and hepatic disease. And then B is saying intestinal obstruction, restless hepatic disease, rectal prolapse. Rectal prolapse is associated with trichosis, tooth grinding, intestinal obstruction, lactose intolerance, growth retardation, and hyperinflation. Hyperinflation is also anyone that knows the actual worm that is associated with hyperinflation. There is actually a specific worm, if not strongyloid, forgotten it specifically. Malabsorption syndrome due to vitamin B12. Now, this malabsorption is vitamin A, not B12. So, in this ascaris, there's going to be vitamin A malabsorption, intestinal obstruction because of the worms. Sometimes it can go to the liver causing inflammation there. Yes, please. C. Tooth grinding, intestinal obstruction, lactose intolerance, growth retardation, and why so? Why do you think so, Doc? All right. So please inform us when you're done. Number 50. Which paired statements is correct about the said parasite? Trichuris, trichuria. Okay, here you need to know this NIH swab, the scotch tape. This is the diagnosis for interrobius. Like where you put a camera tape on the anal area, and then by night when these worms are coming out, then you can remove like that, and then you take it for testing. That's a scotch tape is for enterobius. 51. Associated with hypogammaglobulinemia and respiratory distress. Pneumocystis carine. This guy was thought to be a parasite, but then he's actually not a parasite. Is now pneumocystic gironvi. It's a fungi. This guy causes the reduction in the gamma globulins, which are the antibodies, and it will cause respiratory distress. Pneumocystic geronvi or pneumocystic carine. 52. Regarding Tinea saginata, the scolex is unarmed with four suckers. Remember we said so, isn't it? The armed one was Tinea solium. Can grow to a length of 7. We said this one is up to 15. It's saginata. It's solium that grows to a length of 7. Eggs are highly infectious. Eggs are not even infectious because it's supposed to be for the eggs. No. If the eggs were infectious, it would cause cysticercosis. Adult worms live in the large intestines of human. Yes, please. I've heard the hand again. Is which one? Okay, thank you so much. Yeah. So hyperinflation, please take note. Strongyloids, stachylaris. Number 53. The following can be transmitted via Scolex is unarmed with four suckers. Remember the sodium is the Scolex is armed. This one is unarmed, the Saginata. The following can be transmitted via mosquito bite: Brugia malay, lower lower, Uncherella bancroft. So Uncherella and the Brugia malay can be transmitted. You can see here, these are filario worms. Uncherella bancrofti can be transmitted by the Anopheles mosquito, the Aedes, even by the culex. Brugia malay can be transmitted by Mansonia, which is also a mosquito. And then you also have Brugia timori. Lower lower is transmitted, we mentioned this already, by Chrysops, which is a deer fly. Onchocerca varis is the black fly, Simulium. And then you have got also these others, Mansonella, which is transmitted by the Culicoides, which is the biting midge. Notes method is used to demonstrate the microflora of Brugia and Uncherella. I don't know how I, I've tried to look up the notes method, but the only thing I was saying is that you're going to use it for some lymphatic filaria, for some filario worms, specifically Brugia malay and Uncherella. This is also a filario worm. So I can't say it is only used for filario worms. So specifically it is BW, not BMW. Yes. So you are saying the note. So we are going to put K in front, KBW. The note method is used for Bruga Malay and Wuncherella. Number 55, the most common infective stage of trematodes, Tomanis. So trematodes, these are flukes, these are trematodes. Trematodes, their most infective form is a metasecaria. But it's a bit different because trematodes, you have got different forms. Like cystoma, the infective form is secaria. And you can see you have got cystoma hematobium, which causes urinary cystosomiasis, cystoma mansoni, which causes intestinal cystosomiasis, cystoma japonicum, intestinal also. But for the other forms of the fluke, sorry, yeah, of these are flukes. The other forms of trematodes, the infective form is the metasecaria, like Fasciora hepatica. If you can see here, I've specifically put what they've put in the notes. When I was using Panica's textbook, Fasciora hepatica, they've written there as the sheep liver fluke. But I've put common liver fluke here because that's what they've used in the lecture notes. And this the lecturer is going to mark, isn't it? Unless maybe another lecturer comes and makes that correction, or maybe it's a new correction, the book was lying. Fasciola gigantica, they've put for them in the lecture notes sheep liver fluke. And then Paragnomas westmani is a lung fluke. All these infection is by metasecaria. So most of them is a metasecaria except the cystosome. Yes, please. Oh, yeah. Okay, thank you so much. Now the problem is because in test two they had brought this question, and one question they had said the other name for fasciola hepatica, and then the following question they said the other name for fasciola gingatica, and both answers were there, so it was a bit trick on which one to choose. So in case of choosing, I'll still advise in case it came, you can use the lecture notes slides. If you want, you can go and check Panicas. You'll find it's opposite. They'll use Fasciola hepatica for sheep liver fluke. Actually, even some other readings that I did, that's what it said. But the lecture notes say what I've written here. Hope that is okay. The infective stage of parasite is found in crabs and crayfish. So you can see this guy, the lung fluke, which we've just talked about on top, where the metasecaria is the infective form. Paragonimus wettermani. The parasite, infective stage of the parasite, is found in the crabs and the crayfish. 57. Whipworm infection is commonly in the tropics and frequently observed together with this one someone already mentioned, isn't it? The whipworm. What is the common name for whipworm or the other name for whipworm? The Trichuris trichuria. Thank you so much, Doc. Trichuris trichuria is found together with Ascaris lumbricoides. Just think of the simplest word at Ascaris. Let's hope someone will not think of ancylostoma in case they bring and they've put ancylostoma there. Depending upon the intensity of infection, inflammation may extend from the distal part of the small intestine to the rectum, where the mucosa becomes edematous, and friable. This clinical presentation is seen in. Yeah, so what we need to know about trichosis is that this guy, especially in heavy infection, it can be very, very bad. It can be very bad such that it's going to block these large intestines, and then there can be edema, a lot of edema there. And that is a characteristic feature. And inflammation, a lot of inflammation, especially in when there are a lot of worms. Which of the following viruses are mosquito borne? Remember, we said that the viruses that are carried by mosquito are arboviruses, and we mentioned that chikungunya virus is an example of an arbovirus, which are carried by arthropods. The following symptoms pertain to the whipworm or whipworm infection. Whipworm, we know this is trichosis. And one thing you should always think about when you think of trichosis, what symptom? What symptom comes to your mind? Rectal prolapse, yes. So there is a rectal bleeding, and this guy, as we've said from the top, that inflammation, big inflammation causes the rectal bleeding. That's why they can as well be anemia because of the a lot of loss of blood. And then, so rectal bleeding, rectal prolapse, diarrhea, frequent defecation. So that's the best. Rectal bleeding. The others, hyperinfection, we say that is the strongyloid, so for B. And then C, there is what word there? Crawling sensation. When you hear of crawling sensation on the perianum, what are we thinking of? Anyone? Thank you. Enterobius vermicularis. Rectal bleeding, diarrhea, frequent defecation, nightmares, even nightmares. Okay, that one is wrong. The following antigens is used on a point of care malaria detection kit: CXCR4, PLDH. So I've even put it there, PLDH, which is a plasmodium lactate dehydrogenase. It's an enzyme which is produced by the plasmodium, and that causes, which is the guy which causes malaria. So this plasmodium lactate dehydrogenase is triggers. Uh, is actually going to be used for diagnosis and antigen that can be early detected even in the point of care detection kit. So that is B, PLDH, plasmodium lactate dehydrogenase. Number 62, the infective stage of entamoeba histolytica to man has. So what is the infective form of entamoeba histolytica? Anyone? The cyst, yes. And the cyst has got what is known as a bullseye. Pseudopodia is part of the which morphology of the parasite? The trophozoite. Thank you. How about the ingested red blood cells? It's also trophozoite. So yes. So those are not. It's a bullseye karyosome. The pathogenic stage of Interamoeba histolytica. So this is what you need to differentiate. That was the infective phase. The pathogenic phase is actually the trophozoite. So you have got the pseudopodia, you have got ingested red blood cells. So A and B are correct. So take note, infective form is a cyst. Pathogenic form is a trophozoite. Seatworm infection can be associated with. What is the other name for seatworm? The same guy where children will be rubbing the anal area. What's the name again? Yes, vermicularis, enterobius. So we know enterobius there is a reinfection, right? Where the guy goes back. Actually, look at this creeping eruption is a characteristic symptom. This one that has been mentioned as a creeping eruption is a characteristic for lava migrant, specifically those caused by animal hookworm like ancylostoma. And then for hyperinflation, there is associated with the strongyloids. The ground itch is a symptom associated with hookworm, where these guys have entered, like where the worm has entered, there's going to be a lot of itching. That is the ground itch. Number 65. Regarding preventive chemotherapy for Biwaza in Zambia, it's not necessary if the prevalence is 10% and below. It is given once per year for prevalence above 50. Only health, education, and hygiene are needed for prevalence 10% and below. None. So this is from the lecture notes where I've put here the frequency of preventive chemotherapy for bilharzia. In endemic communities, prevalence of infection with cystosoma 10% or higher. WHO recommends annual preventive chemo with Prasquanta. 10% or higher supposed to be chemo, but A is saying it's not necessary. So actually B. Oh, actually that option is not there. It's supposed to be 10% or higher. And then B, this second part is saying if prevalence is greater than or equal to 50% of persistent hotspot biannual, that's twice a year, preventive chemotherapy with Prasquantal may be more effective. And this B is saying it is given once per year for prevalence above 50. It's supposed to be twice, not once. And so this is also not true. It's not necessary if prevalence is 10% and below. It's not like when biohazard is there, even when it is less than 10%, it doesn't mean it's not necessary. At least do something, do education. And if you remember, yeah, this is one thing that I still remember from primary school in grade 5, grade 6. These people used to come at our school saying, no, biohazard, wash your hands, and such things. I remember that one at least. In Plasmodium vivax infection, schizons rupture to release 8 to 24. We can see in vivax, it is 8 to 24. In ovary, how many schizons are released? 8 to 10. Thank you so much. And then in malaria, 6 to 12. Thank you so much. The following symptoms are seen in enterobiasis. Enterobiasis. What causes enterobiasis? Enterobius vermicularis. So there's going to be anal itching. This guy actually, like enterobius, can wander. It will go to the reproductive system causing discharge, and also the perianal area, like I'm wondering when that is the one that causes even the rubbing. Rectal prolapse is associated with trichuris. Even here, vitamin A malabsorption. Which one is associated with malabsorption of vitamin A? Anyone? Ascaris lumbricoids. Number 68. produces massive diarrhea in a patient with a low resistance. Okay. Cryptosporidium parvum. There is a lot of diarrhea there. So I've put something here. Pneumocystic carine, which is pneumocystic Gironvi now. This guy causes pneumonia, pneumocystitis, pneumonia in people with weak immune system. And then the Toxoplasma gondii, the cat guy they found in cats. This is a protozoa that causes toxoplasmosis, which can cause GIT symptoms, but not massive diarrhea. 68. Protoscolix of Tinea solium is complete with 1000 proglottids, invaginates in the small intestines, evaginates in the large intestines, possesses a rostellum with one row of large and small hooks. So when you look at the stages for tinea, you have got the egg, specifically the egg or the gravid proglottid is going to be passed in feces. Remember the proglottisal segments. And then what happens now? You have got the oncosphere. So when the pigs ingest the eggs, the oncosphere, which is the embryo, is going to be released in the intestines. And there, the oncosphere will penetrate the intestinal wall and migrate to the muscle where they develop into cysticercae. And then the cysticercae, also known as bladder worms, are the larva stage of the tapeworm. They are found in the muscles of the pigs and the infective stage of human. When human consumes undercooked or raw pork containing cysticercae, they become infected, and then we've got the protoscolex. So the once ingested, the cysts are now going to the cysticercae are going to invaginate in the small intestines of the human host. The scolex, that's the head, attaches to the intestinal wall and begins to grow. And then you are going to have now the adult tapeworm. The scolex develop into the adult tapeworm, which can grow up to several meters. The adult tapeworm consists of the head, which is the scolex, the neck, and the series of segments, which are the proglottids, which contain both female and male reproductive organs. So here it is complete with 100 proglottids. Proglottids are found on the, like that's like their tail, not this guy, which is like the head, the scolex. It vaginates in the small intestines. That row, isn't it? In the small intestine possesses a rostellum with one row of large and small hooks. The scolex itself, the head, is the one that possesses the rostrum with one row of large and small hooks. But the protoscolex is a stage, not the head. What parasites may produce Loeffler syndrome? So I've picked Enterobius. What other parasites produces Loeffler syndrome? We did mention them, isn't it? Yes, please. Ascaris, lumbricoids. Yes, thank you. Anyone else? Strongyloides. Thank you so much. Another one. Every time where you see Enterobius, okay, you have got Enterobius here. There's another one that is remaining, I think. Which one is remaining? Or maybe they are the only ones, these guys. Nothing? Okay, so you have got the hookworms. So hookworms, the Neceta americanus and the Ancylostoma duodenale. So we have got hookworms, ascaris, and the strongyloids. So what parasite may produce low fly? Why did I mark enterobius? I'm now thinking twice. So supposed to be ascaris, lumbricoids, strongyloids. About enterobius, it's supposed to be the hookworm. So I think I thought one of the hookworms is enterobius. So for the hookworms, you have got Necata and Ancylostoma. So it is actually supposed to be A, not B. I've even put them down here. 71. Cysticercae cellulose in the pigs result from ingestion of cyst in cabbage, ingestion of human feces with tinea sodium eggs. Remember when the pig ingests the eggs with the uh, through the feces, then it's going to enter and then it will be able to differentiate into the cysticercae in muscle, such that when we again go and eat that pig, we now get this, we develop cysticercosis. Chronic salpingitis may be seen in all of the follow, in which of the following? So salpingitis is the this vermicularis. What it does is that, this guy infects the anal area, but it likes moving. It will go to the urinary tract and then it is going to go and cause inflammation of the fallopian tubes. So that vermicularis is a bad guy. The following statements are correct about nematodes. Remember we said nematodes are segment nematodes, except they are correct. Oh, the following statements are correct about nematodes except unsegmented. Remember, we said they are unsegmented. Their sexes are separate. Some require a host, others don't require a host. That's true. We said they don't multiply in man or in any host. They don't multiply unless they are other worms, not nematodes. These are the properties. I've even put them here so that we can look at them. The species, so adults is an elongated cylindrical that, also known as roundworm, unsegmented body covered by fine, smooth cuticles, sometimes striated, has an inner body cavity. At least they have got the entire GIT, typically different sexes, separate sexes, transmission to a host by ingestion of the mature infection, and those nectaries do not multiply in host. Some require host, others do, and such. Some require one host, others two hosts. The following have been characterized as carcinogens. So some parasites have been classified as carcinogens, like this guy, Clonochis sinensis. Some parasites are now classified as carcinogens, meaning they lead to cancer. Like for example, the liver fluke, Clonochis causes bile duct carcinoma, and also the Cystosomia hematobium that causes urinary bladder cancer. So these guys, they are carcinogens. Please take note of the two, clonus and hematobium. In cyclophilidian parasites, the strobila consists of. Now the strobila, remember the way we talked about a worm? You have got the head, right? And then you have got the neck. And then you have got now the segments. And we are saying the head is a scolex. This is just the neck. This is the strobilus, which is the entire structure. Strobilus has got proglottids. Proglottids, these are segments which can be shed. And you have got up to 1,000 of these. And some of them are mature, others are not. So immature proglottid, the neck is also considered part of the strobilus. And also gravid proglottids, these are the ones that have got the eggs, they are gravid, they are pregnant. Batchman intradermal test and xenodiagnosis are used for the diagnosis of this one. Please just is it swatter? Yes, please, doc. So this shima end of the worm. starting from the neck to the immature proglottids and these mature proglottids. The proglottids being the segments which are shed and then the eggs are released. Each proglottid contains Faustina saginata. Each one of these contains 40,000 eggs. Okay. So the Bachman intrademo and Zeno diagnosis. So Bachman intrademo, this is an allergic reaction test that involves you inject the trichinella antigen into the skin, and then a positive reaction indicates the presence of the antibodies against trichinella because we are injecting the trichinella antigen. Xenodiagnosis involves feeding muscle tissues suspected of containing trichinella larvae to laboratory animals. If the animals develop trichinella infection, it indicates the presence of the parasite. But that is bad. You want your friend to be sick. 77. Regarding occult filariasis, the following is true. So, occult filariasis refers to a condition where the individual are infected with filarial parasites but do not exhibit the typical clinical symptoms of filariasis, like, for example, elephantiasis or lymphedema. Instead, this filariasis is only characterized by massive eosinophilia and subclinical symptoms like non-specific fatigue, weight loss. You see, they don't have those characteristics. So, regarding occult filariasis, the following is true. Isolation of microfilaria is definitive, like you can't get those. Presence of classical filarial features, there is no classical. Wet cough in the night tells it all. This is non-specific and usually presents with massive eosinophilia. Yes, there's going to be a lot of eosinophilia. Mature proglottids of Taenia solium are, remember I said Taenia solium, you have got the mature, the immature, and they're actually square shaped. This take note because immature proglottids are transverse rectangular, like they are bigger in this way. That's the immature. And they are located in the anterior part of the body and inner organs. The mature ones are square here and they're located in the middle part of the body. And I've got 150 to 200 testes, centrally straight uterus, and 3 lobes of ovary. Acute cystosomiasis. Yes, doc. Sure, please. Thank you so much. So, gravid, an addition, longitudinal, rectangular. Okay, thank you so much. So, immature, they are transverse. Mature, they are square. And then the gravid, they are longitudinal, rectangular. Thank you so much for that. So acute cystosomiasis, I've put it here. Acute cystosomiasis or Katayama fever, a serum sickness like syndrome with fever, rash, myalgia, arthralgia, cough, generalized lymph adenopathy, hepatosplenomegaly may develop. Individuals with acute cystosomiasis show high peripheral blood eosinophilia. So mostly present with jaundice. We can say there is no jaundice there. Of course, there is hepatosplenomegaly. When these guys become big, there will be breaking of the red blood cells, but not most. Notoriously caused by cystosoma meconium, which causes more harm than others, can present as pipe stem fibrosis. Is a serum sickness like symptom. Like we can tell, right? That's the most just direct from the notes. Retroinfection can be seen in the named nematode, and I think we've been mentioning this for long now. Retroinfection is in Enterobius vermicularis. So Enterobius vermicularis infections can be the actual infection which occurs in healthy person when they ingest infective egg. There's also reinfection, which is self-infection. A person can reinfect themselves from their own. by ingesting of infective eggs. And then retroinfection, this occurs when the eggs mature and the larvae hatch in the anal area. This larva can migrate back to the large intestines through the anus where they grow into mature worms. That's retro, back direction. Geophagia may, which is also known as pica, may be seen in heavy infection. We already mentioned this one. Pica is associated with, what is pica? Anyone? What is pica? Thank you so much. Craving of non-food substances like eating soy associated with ascaris. Dash cysts are shed intermittently in feces, and laboratory confirmation of the diagnosis often requires a patient to produce more than one fecal sample. Like Giardia rambia, they are intermittently shed in feces. So you need to be presenting samples again and again like that. For example, today you might present a sample and tomorrow you find more than what you saw today, or today you see more, tomorrow you see less. Not because you have become, you are treated. It's because there is intermittent shedding of the cyst. Which among the of these produces Siemens dot? Giardia, option C. Siemens dot. So about the Siemens dot is the Plasmodium malariae. Remember we mentioned of the, which one was that? I said Skufner. Skufner's dots, you are going to see them in ovarian vivax, but they are not present in malaria. Instead, the malaria has got the Siemens dots. So that's one we need to take note of. Following statements pertain to Tinea solium, gravid proglottid. Remember, as we already, I think we already mentioned this. This is Tinea solium. We have got 40,000 eggs, and these guys are non-mortal. Would have picked this, but 25 to 30 many lateral branches is for Tinea saginata. It's I've put here from the lecture notes. So I've got 7 to 10 many lateral branches of the uterus, which we can say as 7 to 13 in that case for Tinea solium. Separate separate from the strobila and passed in feces or migrate to anus. Eggs freed from proglottids. Gravid proglottid contains 40,000 eggs and not mortal. Gravid segment of T solium contains central uterus with less than dozen lateral branches, while gravid segment of T saginata contain central uterus with 15 to 20 lateral branches. So don't pick this one because that's for saginata. Okay. So let's look at this again because I've just realized a doc mentioned something. Contains about 40,000 eggs, non-mortal and longitudinal rectangular. Doc, you said it's a doc that says something about this. So what would you say about this? Contains about 40,000 eggs, non-mortal, longitudinal. Since the doc I think mentioned gravid, proglottid is longitudinal, rectangular, isn't it? So that one, looking at these notes, That's why I've gone to pick this. Though they're not specifically are saying 7 to 10 and this is 7 to 13. Just that I didn't see anything that on the longitudinal rectangle shaped proglotid. But that's what our good doctor does. I think he did research on that. Okay, if you checked, so I was looking at, you can see this one here, 7 to 10 many lateral, and this is many lateral. Because it's less than a dozen, these are just lateral branches. And the main lateral branches, hope that is okay. Opportunistic protozoa in the stomach and intestine. This guy is not a protozoa, so we remove it. Toxoplasmosis, this Gondi guy, not necessarily opportunistic of the stomach and intestine. We get this guy, of course, from the cats and those things. Crypto Cryptosporidium parvum is really opportunistic, and it's of this GIT. Think we've described it a bit, causing the malabsorption syndrome and such. Specimen for the diagnosis of Trichomonas vaginalis infection in female. So female Trichomonas, we know this guy causes these UTIs. You can use urine or the discharge, vaginal discharge. Prostate unless in males. The following statement applies to Plasmodium malariae. So Plasmodium malariae, the infective mature red blood cell, it infects the mature red blood cells, and the sesagon is 72 hours. For the others, most of them is 48 hours. Now, for which one infects the young red blood cells? Anyone? Ovarian vivax. And then, how about falciparum? Which one does it infect? Thank you. It affects both the old ones and the young ones. Most frequent finding in the infection caused by the parasite is persistent hyperinophilia. We did mention this again, isn't it? Hyperinophilia, which is persistent. Toxocara canis. We did mention this again, I think. So there is a lot of neutrophils in this condition caused by Toxocara cani. You are a physician in OPD and you receive a patient with symptoms of malaria who alleges that his feelings cannot be caused by anything else besides malaria. You do a test and it comes out negative. The following might be the reason for the negative test. Antibody are not yet formed, hence the negative test. The parasite are at the liver stage, which cannot be detected. IgM antibodies concentrations are still at low concentration. Remember we said that when the parasite is in the liver, you can't detect it unless it goes into blood. Cytoadherence in Plasmodium falciparum infection can be enhanced by the following ligands. And we did mention, right? We did mention VCAM1, ICAM2, E-selectins. So except this one is not part of those ligands that help in cytoadhesion. Which of the following shows dual periodicity? Lower oncoseca vulgaris, vulvarus, Manzanella streptosica. Lower lower, this African eyeworm exhibits dual periodicity. This means that the microfilari of lower lower present in the peripheral blood during the day and migrate to the lungs at night. And this is because, like in the blood during the day, because of, remember the fly that causes the infection for lower lower. This normally bites during the day. So if it bites in during the day, the microfilarial have been injected, and then at night they'll now go and hide in the lungs. The following statements apply to Taenia solium. Single proglotid contains two except 200,000 eggs. We say this is not true. Single proglotid contains 40,000 eggs. Neurocysticercosis is possible. Yes, being caused by the larva, right? And then ocular cysticercosis is possible, specifically in the anterior chamber where we've got a whitish grayish mass. Tissue invasion is relatively slow and tends to stimulate granuloma formation. Acanthamoeba, castellani, Balantidium coli, Interamoeba histolytica, Naegleria fowleri. When you look at this, the primary amoebic encephalitis can be caused by Naegleria fowleri, which is the brain-eating amoeba, where someone is swimming and then the Naegleria enters via the cranial nerve number 2 and then it goes to the brain. And then Acanthamoeba can cause granulomatous amoebic encephalitis and chronic amoebic keratitis. So we're talking about granulomatous, which occurs slow, relatively slow. Acanthamoeba. The Interamoeba histolytica also forms the granuloma, which is an amoeboma. But from the notes it says it's quick, fast. So this granuloma for acanthamoeba is slow. Why does Palantidium coli cause? Anyone that has read on that? Right, thank you so much, dysentery. Which statement is correctly paired? Plasmodium vivax, benign quartan malaria. We know Plasmodium vivax is, we want what is correctly paired. Plasmodium vivax and ovale, which one do they cause? It's a benign tertian. And then C is saying Plasmodium malariae malignant tertian. So Plasmodium malariae causes which one? Is this one, which is even picked, isn't it? Benign quartan. Which one causes the malignant tertian? Fa- Falciparum. Thank you so much. The following is a vector for Leishmania. Sandfly, mango fly, black fly. So is sandfly. Which specific genus of the sandfly causes Leishmania? Anyone? It starts with a G, right? The Glossina, thank you. So the mango fly, chrysops, these ones are for lower lower. And then the black fly, specifically Simulium, is for onchocerca. 96. The following statements apply to Tinea saginata except an armed tapeworm of man with about 200 proglotids. 200 is wrong, right? The eggs are non-infectious in human as neurocysticercosis is not possible. So neurocysticercosis, the eggs are not infectious in human. The following statements apply to teens except. So this one, the eggs are non-infectious. Why do you think this statement is not correct? Anyone? So what is non-infectious to human for Saginata is actually the larva, isn't it? That's the one that causes cysticercosis, not the egg. Ingestion of eggs by human can end up with larva form in the brain. Again, this one, ingestion of eggs can end up with larva form in the brain, eyes, and muscles. So I'll come back to it. And then D, cattle becomes infected by grazing or pastures polluted with human feces. This one is true, isn't it? It applies. This is true because what did I even mention? So I said I'd confuse that. The Tinea saginata, the larva, is the one that causes cysticercosis. But that larva, it's actually still, it has before, it's found as it's known as what? Oncosphere. It has not yet broken out. So it's still as an egg, it's in the egg. So this egg can't infect human. That's why you can't have neurocysticercosis. That's a larva form. And then an arm tapeworm of man with 2,000 proglottids. This is specifically supposed to be 40,000, but the 40,000 is more specific to Tinea solium. What I was finding was for this Tinea saginata, like 2,000, it is 2,000 to 40,000, like something like that. So look at, see, ingestion of eggs by human ends up with the larva forms. So these guys don't cause any infection. It is even more specific. It doesn't. The eggs though don't cause infection for Saginata unless the adult worms. Remember the eggs and the larva; those are babies. Babies don't cause infection. So that's why this is totally wrong. The 40,000, this is per proglottid, specifically for Saginata, for sodium, not Saginata. Regarding cockroaches, the following is true. Usually indicators of deteriorating infrastructure are bad because they are associated with bacteria only. Both mature and immature ones have wings. Insecticides used in is the only method for their elimination in established cases. So this is true. Where there is deteriorating infrastructure, you're going to see cockroaches are bad because they are associated with bacteria only. Even the word only, when you see it normally, is wrong. Both mature and immature ones have got wings. Like the immature ones don't have wings. Insecticide use is the only way of eliminating. How about beating them? Isn't it? Can't you kill them? 98. Mosquito vector of malaria in Zambia. So Aedes aegypti, this guy is associated with the transmission of arboviruses like chikungunya and Zika. Anopheles flavirostris, this one there's no direct one like that in Zambia. So from the source I use this now, it's not there. Colexi cucufasciatus, this one is associated with Oncherera and Bancroft and West Nile virus. Anopheles gambiae Ciso stricto. It's the one that you are going is going to cause mosquito. Together with what funestus, house flies are important agents of the following. So house flies act as chemical arthropods. The chemical ones, these are guys, arthropods can be classified in this chemical. You have got the vectors. So the chemical ones, these are the ones that can just come and touch one thing and then they go and infect another portion that can cause infection. So for example, there is dirt somewhere. The flies can go and touch there. Whatsoever parasite is there, they can carry it. They go and step on the food. They will leave it on food. That's how it spreads. So Enterobius, Giardia, Trichinella, any parasite. The following parasites inhabit small intestines except, remember again what we said, small intestines. If it's not Trichuris and Enterobius, these are the only ones for large intestines. So Enterobius is the answer because the rest will inhabit small intestines.